Good afternoon and a warm welcome to all attendees of this EMA EORTC workshop on the role of patient-reported outcomes and health-related quality of life in regulatory decisions. From its inception, this workshop has evolved with ambitious goals, and it's a pleasure to see so many of you here today. When assessing treatments as regulators, we often grapple with choosing between precisely quantifiable pharmacological effects and the broader but more challenging to measure impacts on health and well-being. Today is a great opportunity to look at strategies for bridging this gap and to explore ways to optimize regulatory decisions. Without further delay, I'm delighted to pass the floor to my co-chair, Madeleine Pey, head of the Quality of Life Department at EORTC, who will tell us agenda and objectives.
So I just want to say thank you so much, Francesco, for that warm welcome. And it's also a privilege for me to welcome you all to the EMA EORTC PROs in Oncology workshop. And by training, I'm a methodologist, a statistician with a special focus on psychological outcomes. So this kind of workshop is really close to something that I appreciate. So I'm sure some of you are aware that this is not the first time we plan to do this workshop. So this workshop was originally scheduled in March 2020, the day before the lockdown during the COVID-19 pandemic. And so 4 years later, here we are, thanks actually to Francesco Pignati and to Danila Com, who kickstarted this initiative and provided us all the opportunity to gather today to talk about the use of PROs in oncology for regulatory decision-making. So Caroline, Volt, and I were then tasked to coordinate and develop this agenda together. And when we were planning today's workshop, we started with revisiting the sessions and the presentations that were planned back in 2020. But really, so much movement has happened in the PRO space since then that we had to rethink the agenda and make sure that it reflects the advancements in the field over the last 4 years. So just to name a few of the key initiatives that occurred, we have the standardization efforts such as Spirit Pro, the Consort Pro, the CISACOL, Proteus. We have the EMA Oncology Working Party Strategy Plan and the recent activities of the EMA on patient experience data. We have the FDA series of guidelines on patient-focused drug development and the use of PROs specifically in oncology drug development. We had the release of the UNETA endpoints guideline, which is now HTAR, and also the plan of ICH to develop guidelines on patient-focused drug development. And probably I have missed some more initiatives, but really the purpose is to provide you with a snapshot of how quickly we have evolved in the past 4 years. So we went back to the drawing board and developed an agenda that would fit with the growing movement in PROs in oncology. So we could easily fit a 2 or 3 day meeting on PROs, but the reality was we had less than 1 day to fit all of these topics. So we had to make some really hard decisions, but it also allowed us to focus on what we want to achieve in this workshop. So we decided that rather than having one presentation after the other, we wanted to highlight key presentations that would resonate across various international stakeholders and provide more time for a panel discussion with the goal of developing concrete action plans by the end of this meeting. So here we are today to discuss patient-reported outcomes, health-related quality of life, a topic that is very relevant to all our stakeholders, but most especially to the patients, because this is about their life and their reported experience. So this platform is an opportunity for an international multi-stakeholder discussion to concretely identify how patient-reported outcomes and health-related quality of life can inform the benefit-risk assessment of cancer treatments. So we are immensely grateful to all our chairs, speakers, and panelists who agreed to contribute to this workshop. And they are the main actors in driving the ideas forward and stimulating the discussions that we will have today. And I also want to say thank you to Monica Turek from the EORTC, to Charlotte Halin, and to Guillem Mathieu, I hope I'm saying his name correctly, for providing all the support for the organization of this workshop. So with that said, let's get started. And I give the floor to the chairs of the first session, Jan Bogaerts and Caroline Volz.
Thank you very much, Francesco, and thank you very much, Madeleine. So it's with great pleasure that I'm co-chairing the first part, which is an introduction and meeting objective of the quality of life workshop. So my name is Caroline Volz, and I work in the Oncology and Hematology and Advanced Therapies Office. And today's first introduction will be by different speakers and setting up the objective of the workshop. Maybe I can also give the floor to Jan to introduce himself.
Good afternoon. My name is Jan Bogaerts. I'm the scientific director of EORTC, and I'm also somewhat active at EMA on the SAC-O. I want to thank Caroline because I was there when I saw Madeleine and Caroline do an enormous amount of work to make all of this possible. So thank you, Caroline. I give it back to you to introduce the first speaker.
Thank you, Jan. And our first speaker is Pierre Desmoulins. So Pierre has been long in the system. He's the chair of the Oncology Working Party. He was also the vice chair of the CHMP, and he's the vice chair of the Scientific Advice Working Party. So it's with great pleasure and honor that we start this meeting with Pierre. So Pierre, over to you.
So thanks a lot, Caroline, for those nice words. As you could guess from the accent, I'm from the French agency on the basis. But it's a big pleasure to have that presentation today. And it's a bit intimidating to be the first one speaking in the afternoon. So I trust that you had a very good lunch and you won't listen too carefully. So it's my only hope. It's my only hope today. No, I'm giving you a speech, so don't be afraid. It appears a bit negative at the beginning, but it will end positive, like any successful movie. So I hope it will be successful. So let's have the first slide, please. I was asked to say my opinion, to give you my opinion on what exactly is the patient's perspective in terms of oncology. And I believe that oncology is a very special field here. Not because I'm very interested in oncology, but because I believe that oncology has this originality of being a polyphasic disease. Not the only one. But let's have a look at an example. A patient with a diagnosis of cancer will possibly get adjuvant or neoadjuvant treatment, could be cured, but if the treatment is not successful to prevent relapse, after a period of silence, the disease will come back. And M1 here symbolizes the first metastatic or advanced stage of the disease. Not all patients go through this, but some. And some start the disease at that stage. But after that first metastatic step could come a second one, after usually a shorter time without treatment or without disease, not necessarily. And then M2, M3, and finally the outcome. Not all patients die from their cancer. So every patient has a different story. Not only because every patient has a different story, but because any cancer is different from any other cancer. If we're speaking of prostate cancer, we're not telling the same story as for pancreatic cancer. All of you know that very well. So let's go a bit further. And something we are not discussing here, but which is a good example to me, are patients' preferences. It's very interesting to ask patients, what would you value in the outcomes as something which is important to you? Would you value nausea for 2 weeks at the same level as the hope of getting the disease coming back 2 weeks later? Just as to give you an example. But the question is, who is the patient we are asking this question? Let's imagine that we are interested in the adjuvant neoadjuvant setting. We have a lady who is ready to go for the treatment. She was discovered breast cancer 2 weeks ago. And we are proposing a neoadjuvant treatment. She's not experienced in cancer because she has very few symptoms. She's not experienced in the side effects of the treatment. Does she know really what nausea under treatment means? Probably not. She has an idea. So let's ask the question a bit later, after the adjuvant treatment. And here we will have an experienced patient, but the hopes for not getting a relapse could influence her perception of the weights of the preferences. So let's go a bit further. And these patients, after 10 years, is considered cured. She will probably value the problems related to the treatment much less as opposed to a patient who will have relapsed early. And forgive me, patients we will never ask the questions are the patients who died from the cancer. So just to illustrate that when we are speaking of the patients, we are not speaking of one patient. We are speaking of many patients, and even from one single patient being different from him or herself, because we are not answering the same way, the same questions, depending on where we are in the course of the disease. The next slide, please. Does this prevent us from asking questions to the patients? And here we are closer to our topic today, which is quality of life. No, it's very interesting. But what are the questions we are interested in and the answers we are interested in? Let's go back to that patient with her neoadjuvant treatment. Are we interested in the quality of life during the treatment? Are we interested in the quality of life during the neoadjuvant treatment, including surgery, because neoadjuvant treatment could influence surgery? It's very interesting to know what are the consequences of surgery on her quality of life. And then the silent period could be affected by delayed side effects. We hope that she will not suffer from her disease any longer, but who knows? But maybe we should extend the period for the quality of life. And why not quality of life, including the first metastatic step, if it happens ever? It would be interesting as well, because the initial treatment could have consequences on the way the second treatment is tolerated or accepted by this same patient. But we could have exactly the same question for progression-free survival. Are we more interested in PFS, PFS1, PFS2? Is it overall survival? That is the ultimate question. I'm not suggesting anything very original here, but just to underline that quality of life is something we must be very clear on. What are we interested in? The quality of life during treatment, after treatment, during and after, at later stages, and so on, and what are the types of side effects we are interested in. The next slide, please. And then we may have some other questions, because, okay, the disease could affect the quality of life of the patient, and the treatment could affect the quality of life of the treatment. And this is something which is not really specific, but very important in the domain of cancer, because we give some treatments at the maximal tolerated dose. So we know that they will be ill tolerated in advance. And we could imagine that at the beginning of the treatment, especially when the patients are free from symptoms at the start, they will suffer mainly from the treatment. And many patients will say, oh, why have I accepted that? Because I was much better before. And then will come, if we are not successful, the periods where the disease will produce symptoms and will influence the quality of life. Quality of life could be positively affected by the influence of the treatment on the disease, could be negatively affected by the influence of the treatment on the patient, could be negatively affected by the influence of the disease on the patient. So it is not only polyphasic, it is something which is multi-component. And these components are not necessarily simultaneous and change their importance, their relative importance will change over time. Next slide. So what is the question we're asking? And here I have some typical questions that could be questions from regulators, but questions from a physician or questions from a patient to himself or herself, is a 3-month increase in treatment-free periods worth it? It meaning the side effects of the treatment. And this is a question. The 3 months increase in treatment-free period is PFS. And the question is more a benefit-risk question. And the question the regulators are asked to answer. But even if we believe that the answer is yes, it's worth it, any patient or any physician is free to consider that it's not worth it for this single patient. Is the maintenance delaying the next painful chemo by 3 months worth it? EFS is the endpoint. Benefit risk is the question. Is adjuvant treatment increasing cure hope from 80% to 90% worth it? And here we see that we can play with the numbers. Increasing from 80 to 90 is very different from increasing from 20 to 30. It's still a 10% difference. So the cure is the endpoint, and the benefit risk is the question. And we see that here we could have 2 components for the same treatment. The adjuvant treatment can delay the relapse or can increase the cure rate. 2 endpoints that are by nature relatively different with different consequences on quality of life. And the question which is more a question which is within the dialogue from the physician to his or her patient and back. Do you accept a last painful chemo that may bring a few more weeks to your life expectancy? And this is individual trade-off. But to give an answer, the patient must know what could be the consequences of the treatment and how get the information from the experience gained with other patients. And here I come to positive aspect of my talk. The next slide, please. It's not because it's difficult that we should refrain from asking, but it is important that we know exactly what is the question we're asking. It is a problem of estimates. And here I'm invading a field I'm not a specialist of, but it is a matter of estimates. What exactly do we want to know? It's not what is quality of life. It is what is quality of life under treatment, provided that the patient takes the whole treatment for the whole duration of, and so on. So it could inform important regulatory decisions. I'm not saying that this should be the primary endpoint in any trial. For sure, I'm not saying that. But it should be an endpoint as soon as we believe it's important. When is it not important? The answer is never. So we should explore quality of life as often as we can, which doesn't mean that we should just say that. We must be more precise. One question for a regulator: should the results of a quality of life exploration go to the product information, be part of the indication and so on? It depends, but it's not the thing we are discussing. Every information is relevant to the benefit risk discussion for, I'm here taking my heart of a regulator. We discussed the robustness, the relevance, the utility of the information which is provided, but any piece of information must be taken into account, not necessarily to build the whole decision, but sometimes to be simply supportive. If you do not have a strong indicator that the quality of life is a catastrophe because of your treatment, it is an information you can give to the patient. This will not destroy your life and could bring something which is evidenced by the clinical trial, by the comparison within a randomized clinical trial, as an example. It's not because it's not perfect. It's not because it's difficult that the information is not interesting. So this is important. And the whole workshop this afternoon, and I completely agree with Madeleine, it could have been a 3-day workshop and even a 1-month workshop, but the whole topic is that one. What do we do? What do we do with what we get? And what are the consequences? And this is difficult, but let's be the best to have the information as reliable as possible, as informative as possible, to make the most sensible decisions we could bring to physicians and to the patients. We regulators, forgive me, I'm a regulator. So this was the last one, I believe. Was it? Yes. So thank you. I tried to be short.
Thank you, Pierre, for this very insightful and thought-provoking comments, and he lays out in front of us what our challenges are in talking about quality of life and patient-reported outcomes. It's now my great pleasure to introduce Winnet van der Graaf, who's a previous group chair of the EORTC bone and soft tissue sarcoma group. She's very close to home because she works for Dutch NKI. And of course, she is the current president of EORTC. And it is very fitting that she will talk about why improving patient quality of life is part of EORTC's core mission. So how committed EORTC is to this mission. Thank you, Winnet.
Thank you, Jan, and thank you also to the organizers who organized this very important meeting today. And I'm happy that COVID is over for many, many reasons, but this is one of it. I would like to talk today about some history from the EORTC, and maybe there are things I will mention you know already for ages, but it may not be the case for everyone. So going back to the mission of the EORTC, where we really have the statement that we want to increase cancer patient survival and improve their quality of life. And how do we want to do that? Well, we would like to have and generate robust medical evidence through our clinical trials, of course, which should be multidisciplinary, clinical and translational, and lead to progress and new standard of treatment of care. So we really would like to do the practice-changing research that leads to changes in the guidelines and improve outcome of patients. And for that, we need to set standards and being a reference for methodological research and an authority in establishing the standards of treatment in care. For this, we need a multidisciplinary approach. So the EORTC aims ultimately to increase patient survival and quality of life assessed by testing new but also available strategies and compare them with each other and finding out the best strategy for patients. And for today, it's also very important that we help develop new drug and approaches in partnerships with the pharmaceutical industry, together with patients, and all in the interest of patients. What are patients' best interests? We can tell that, but it's like the previous speaker was saying, very different for different patients. So of course we need a treatment that is active, and we need a treatment where safety is better than toxicity, but we have to assess it in the clinical trials. But best interest of patients also mean that we know what the treatment, how this impacts on patients' daily life, including health-related quality of life. And this may depend much more on the treatment alone. It may depend on the gender, on the age of patients. We know from clinical practice that patients from different ages have different wishes also for outcome of clinical trial and also assess life in a different way, where the patients have already other diseases which affect their quality of life. A low income may also affect your quality of life and also maybe your health-related quality of life, support from family and friends, the place where you live in the world, and also whether you have access or whether you have drive for hours to come to hospitals. It all affects the whole interest of patients. So there is a notorious mismatch. Doctors, despite their extended ears, I would say, don't listen well. And they don't know very well what the impact of cancer and its treatment is on patient daily life and the impact on health-related quality of life really is. And it's not because they are not interested, but we also should be very, we should have a realistic view that time is limited and really asking all these details during clinical visits where you also have to organize the clinical trials and the treatment prescriptions. Well, there are time limitations and we have to be realistic. So we need the patient's voice, and that's really very important. You see it here. And at EORTC, we have a patient panel where we really have their input in all clinical trials. Already long ago, we realized the relevance of the voice of patients and patient-reported outcomes, and we really know that there is a balance between the objective outcomes, such as disease-free and progression-free survival, overall survival, response rate, and adverse events. But we need also a more holistic view in how to assess the patient perspectives, because that can give an important additional information to evaluate benefits and risk of the interventions. And therefore, we need patient-reported outcomes. And the most important thing is that patient-reported outcomes come directly from patients and no interventions from doctors or nurses who are also commenting on how they see how patients feel. It's really from the patients themselves. So it may be on symptoms, on perception of daily functioning, but also on their health-related quality of life. And for that, we need instruments which we call patient-reported outcome measurements. Is talking about health-related quality of life new? Of course not. That's what we all know, but it's difficult how to assess it. Already in 1986, so almost 40 years ago, the EORTC Quality of Life Group realized that a research program was necessary to develop an instrument that could assess quality of life for international clinical trials in oncology. And at that time, only a number of studies, mainly in breast and lung cancer, and also one in sarcoma in 1982, had incorporated quality of life aspects. And the instrument to be developed should have a core question and also an option for a modular approach. So we all know nowadays the EORTC QoQ C30. This was published already in 1993, led by Neil Aronson, as you can see here. And the number of citations is increasing every year, and it has now over 48,000 citations. And here you see all the different languages in which this questionnaire has been translated. And it's interesting, I just looked at one of the older publications. This is one from 1996, where the author is writing how he is assessing quality of life at that time with this new instrument, and where he also has an interesting observation. He measured quality of life before and during chemotherapy, where in the abstract it's mentioned some parameters improved while others did not, preventing a simple interpretation. I would call this a mixed response, and I come back to that later. And also that there are arguments for compiling indexes of quality of life while retaining measures for individual parameters and the desire for a consistent international use of an instrument such as this EORTC QoQ C30. So it's interesting to see a bit also the hesitation and also, well, we will need to use it, but how do we need to use it in daily clinical practice at that time? And maybe there's a bit of a contrast to that, and I will come later to that. Today, other questionnaires will be discussed. There are two other core instruments, the EORTC QoQ C15 for palliative care, and the QoQ F17, which measures also functional skills, global health status, and quality of life scale of the EORTC QoQ C30. So only 17 questions here. Many modules have been made ever since, and there are validated questions in the item library. It will be discussed later today. And as said, there are many translations of these questionnaires. So back to the 90s. It's interesting to see that well, we had a need of assessing effects of clinical treatments in an imaging way. and where we had WHO criteria for a long period of time, there was a need to do it a bit more simpler and to do it in a different way. And that was, I will not go into detail, how RESIST was born. In fact, in 2000, there was the publication of Patrick Terrasse, who was at that time director of the headquarters at EORTC. Interestingly, that was very quickly adopted into operation in clinical trials under EORTC auspices, US NCI and NCI Canada Clinical Trials Group. And the whole field like to use this RESIST evaluation, so about imaging. And later on, this was adapted, and we all know the current version from 2009 already, led by Elizabeth Eisenhower. But still, questions remain there, of course. New functional imaging, we see more and more nowadays. The immunotherapy came, but we also needed a new assessment, the iRESIST. Radiomics, what's their place here in the evaluation in an imaging assessment? And the meaning of mixed response, I come back to that. How can I explain to my patient what the meaning of mixed response is, and why would I understand what's happening with a certain treatment when you have mixed responses? We take it still a bit for granted in RESIST evaluations, but as I said, it was seen as maybe an obstacle in assessment of health-related quality of life, where of course you have mixed answers to different individual items. Still, the main question remains how best to evaluate the benefit of clinical trials for patients, what's in their best interest. I would like to discuss one paper with you. It's a paper from the Kingston group led by Samuel, published in 2022, where they gave a nice example of a complexity of endpoints. The objectives of their study were to study if cancer drug trials that show improvement in overall survival or PFS also improve global quality of life in patients with cancer compared with a control treatment arm, and to assess how unchanged or decreased quality of life outcomes were reported in trial publications. It was a retrospective study. They used all the phase 3 randomized clinical trials which reported also quality of life and were published in 2019. And in total, 45 patients with almost 25,000 participants were included. And the results showed an improvement in global quality of life with the new agent was reported in almost a quarter of the randomized controlled trials. And those with an improved quality of life were also more likely to show an improvement in overall survival versus trials with an unimproved quality of life, as you can see here. 6 trials reported a decrease in quality of life, and interestingly, 3 of them were with targeted agents. I think targeted agents were always seen as those trials which don't affect your quality of life that much. It's the old chemotherapy, which is horrible. But you can see here that maybe not, well, it's an example again that not toxicity grade 3, but also grade 1 or 2 can affect quality of life a lot if we look at targeted agents, but also we have to look at the target, of course. 11 trials reported an increase in quality of life, and interestingly, more than half of those were with immunotherapy drugs. And then finally, of the 34 trials in which quality of life was not improved, they saw that almost half of them reported these in a bit of a positive frame, like maintaining quality of life can also mean that there was no improvement, things like that. So in conclusion, only a small proportion, a quarter, showed benefit in global quality of life, which had an association with OS, but not with PFS. Well, it all comes back where EORTC was saying we want to do trials which have outcomes that are in the best interest of patients, and that aligns with a new initiative that was launched last year in Kingston, Canada. And I was happy to take part in this initiative with other people here in the room and online, who were also there. This initiative was led by Chris Booth, who will be in the panel later today. And in fact, there the vision was that if we do cancer care and clinical trials, we should really focus on outcomes that matter to patients. And I put two important aspects here for clinical trials, that clinical trials should really be meaningful to patients, and we should also have those assessments in clinical trial approval. Outcomes really matter to patients. We should collect data in clinical trials and make objective, relevant assessments of patients' health-related quality of life next to imaging and survival endpoints to serve our patients, but also, of course, and that's why we are here today, the regulators. And the long history of clinical trials, data collection and input from our patients panel and experts in all the disease-oriented groups and task forces and the headquarters at EORTC enable a better insight into optimal trial design and analysis. And with that, I would like to thank you for your attention.
Thank you very much, Winnet. I personally really appreciate the juxtaposition of RECIST on one side, where we take RECIST for what it is, and that's telling us a lot, and quality of life and patient-reported outcomes on the other side, where we're not necessarily giving it the same breadth of interpretation, and that's something we're talking about today. So very fittingly, the next speaker is Jaap Reineveld. He's an active member of the EORTC brain tumor group. He's also very close to home to Amsterdam UMC. And of course, he's the current chair of the EORTC Quality of Life group. And he will talk to us about what we are still seeing in the much more prevalent quality of life outcomes today. And it's a bit tongue-in-cheek, I believe. It's a tale of two trials, improving the use of PROs and health-related quality of life in cancer clinical research.
Yeah, thank you, Jan. It's a tale, it's not a fairy tale, actually, to make it things clear, because it's about two trials in a very devastating kind of tumor, which is a glioblastoma. But I'll come to that later. First of all, thanks everyone for making this possible. As already mentioned, we tried to do this 4 years ago, but at that time, this was the way I saw my group. Fortunately, we're back to normal now, and this is the way I like to see my group. And this is not my group, it's our group, and I'm very privileged to be the chair of the EORTC Quality of Life group, which is there for 44 years now, and we have a very active membership of over 500 people, and it's really an honor to be there. Well, let's go to the tale. This is about two trials who were both published in a journal you probably know. At about the same time in 2014, they're both on the same patient population, newly diagnosed glioblastoma patients with an average survival, overall survival of about 13 to 14 months, which is really devastating. And of course, everybody, I think they were even in the same issue of the New England Journal of Medicine, and everybody, of course, compared them. And there was a lot, in a lot of ways, they were comparable. They had exactly the same patient population, as mentioned before. They had the same treatment, which was the standard treatment, which consisted of temozolomide, chemotherapy, and radiation therapy, whether or not combined with bevacizumab, Avastin at that time, or placebo, double-blinded. The sample size was a bit different. The European trial, which was the Chino trial, was a bit larger than the American trial. And unfortunately, it turned out that the overall survival didn't increase by the experimental treatment, while the progression-free survival did a little. But then we looked at the patient-reported outcomes in both trials, and they had opposite conclusions. Worsening in health-related quality of life was the conclusion in the American trial, and improvement in quality of life was the conclusion in the European trial. Of course, it can be a transatlantic issue, but it was not very probable. So what went wrong there? And we asked ourselves, and Madeline actually asked the questions, so I thank her for that. The questions, 4 different questions. Were they assessing the same patient population? Were they assessing the same health-related quality of life areas at the same time points? Were they assessing the same endpoints? And were the same populations of patients included in the analysis? And I will go through that, and then you might also get an idea of what you get when you ask for patient-reported outcomes as far as the EORTC is involved. Well, were they assessing the same patient population? No, I think basically they did. Newly diagnosed, histologically verified patients with a certain functioning scale. So that was not the issue, I think. Of course, we know that there is a selection bias. The clinical trial patients are not the real world patients, but that's another discussion probably. And there might be some cultural differences, as said before, between the American patients and the European patients. And they might score differently on several quality of life related items, but that's about it. So that was not the biggest problem. The second question was, were they assessing the same areas at the same time points? Now, and they both used the EORTC measurement vision, and Johannes will present more on this later on, but basically we have a system of the C30 module already mentioned by Rinette, added with a specific disease module, and depending on the issues that might come up because of a certain treatment, for example, we add items from the item library, which consists of validated questions. It's all about 1,000 questions or more, I think, and that's the measurement system. And we have the system that is free of charge use for academic users, and we ask the pharma to pay royalties, and through that income we update our portfolio constantly. The development of such a module is quite a process. We have now, as I remember if I remember correctly, 32 validated questionnaires, and this is a process that easily takes 4 to 6 years, starting with a collection of items and issues that might be relevant to patients, then putting them in traceable issues, and then do a first study in a smaller group of patients and ending up in a field study. And so I think we should say that the patient voice is already in there. There are at least 500 and very often more than 1,000 patients who have added to developing a specific EORTC questionnaire. So we have the idea that at least most questions are relevant because of that. Well, what did they use in the 2 different trials? They use the same questionnaire, the C30 and the Brain 20, which means 30 general questions and 20 questions specifically for brain tumor patients, developed both questionnaires through the same system, but they use different primary quality of life outcome measures. One used the cognitive functioning as the primary outcome, and the other one used the global health status. So in that way, it was already a bit different. Well, it also depends on the timing of PRO assessments. This is one of the EORTC studies I reported on, and luckily we got it in Lancet Oncology, and even David is in the room, but he didn't notice that we had quite a flaw in that one, because this is a comparison of 2 different treatments, and which were not blind, because one of them was radiotherapy and the other one was chemotherapy, so it's pretty difficult to blind. And the first measurement was at 3 months, and that was in the middle of the temozolomide chemotherapy cycle for half of the patients, but already 6 weeks after the last radiation treatment for the other group. So it also matters when do you measure, because that was quite an unfair time point, so to speak. So that is also important. But the time points were quite the same in both trials of my tale. Were they assessing the same endpoints? That's the third one. Well, the one trial used the change in quality of life score at 46 weeks, which is about 10 months as an endpoint. And the other one used the time to a more than 10-point worsening from pretreatment scores that both had a baseline score, obviously, for quality of life as well, or disease progression or death. So the two trials were responding to different aspects of the data. And why is that important? If you see this graph with two different hypothetical treatments, the red one and the blue one, it's quite a thing, because when you look at the time to deterioration analysis, then the red treatment is much better. When you look at the overall analysis, there's not really a difference. And when you look at the end, at the treatment at 42 weeks, then the blue treatment wins, so to speak. So it matters, the choice of your endpoints matter. Okay, one back, sorry. Yep. So, and it also depends on how you handle missing data. What if a patient's disease progresses and the patient doesn't respond to the question, to the questionnaire at week 46? Do you ignore it, or do you imputate data? And they handled it differently. So there's quite a few changes in that one. Sorry. And were the same populations of patients included in the analysis? That's the fourth question. Well, we know that that compliance has different aspects. There's a compliance on patient level with better functioning patients being more compliant, obviously, but there's also even an institutional level we noticed through several EORTC studies that institutions with good quality of life compliance have better survival outcomes and vice versa. So that also are important aspects. Well, what about the analysis populations in both studies? In one study, there were only the analysis population consisted of all patients alive and free of disease at 46 weeks. And in the other one, there's all patients included in the trial, which is quite a difference. Well, so the quality of life results of those two trials are not directly comparable, although they looked like they were. And that is important. And I don't want to only show what is wrong, but maybe also some possible solutions. And when you look at those questions, well, the first question is really, it's a major thing, but it's not on PROs, I think. Every trial design should think about the patient population and about the comparability of the patients. Were they assessing the same quality of life areas at the same time points? There we have a chance to intervene. And that is what we do with our group. As mentioned before, we have a system of the modules and the generic C30 and the item library, and we really put effort in developing validated questionnaires and keeping them updated. But that's not all. We also try to teach the disease groups within the EORTC to use them and to use them properly. For example, think about timing of assessments and things like that. So we will constantly try, and the collaboration between the quality of life group, who was a bit on an island a couple of years ago and now more back in the main community of the EORTC, tries to help the disease-oriented groups and vice versa on that. Regarding the last two questions, that's a thing that's also beyond EORTC. Madeleine already mentioned the different guidelines for example, reporting, designing and reporting PRO data. But one of the things that was missing, that we also realized because of the comparison of these two trials, was that there is not very much uniformity about the statistical methods for analyzing PRO data. And for this reason, the EORTC started the CISAQoL consortium, and CISAQoL stands for Setting International Standards in Quality of Life Research. And we have an almost global group of both regulatory bodies, including EMA and FDA and several others, medical institutes, industry, and academic societies, as well as hospitals. And we've been working on this starting, I think, in 2016, funded initially by a European Union Horizon grants, and now through other funding bodies, to define a set of standards, starting with definition of endpoints and everything following that, so that we really will have a framework how to analyze data, PRO data of clinical trials. And I think the CISAQoL project, and I'll look to Melon, will be finished in 1 or 2 years from now with a set of standards, but it's work in progress all the time, I think, and it's already been published, part of it. So basically, to give you some messages to take home, our group, and it's a bit of an advertisement, apologies for that. We have an extensive portfolio that might help you. We do not only build planes, so to speak, the question is, but we also try to teach people how to fly with them. And the next challenge will be to assess how to incorporate those measures in the regulatory decisions. And that's what we're here for, I think. Thank you very much.
Thank you. Yep, I think it's always very interesting to hear a practical example and how methodological aspects are also when looking at and interpreting different clinical trials. So our last speaker for the introduction is Dr. Juan Garcia Burgos. So he's working as Head of Public and Stakeholder Engagement Department at EMA. And he will provide his presentation on the experience so far at EMA from involving patients and the journey since the very beginning of EMA. Thank you, Juan.
Thank you very much, and good afternoon, everyone. It's a pleasure to be here, and I would like to thank all the efforts to have this discussion today. I think it's great that despite the pandemic and the discussions 4 years ago, you have managed to still put it in place. And I will give a presentation trying to give an update on where we are with the initiative of patient experience data, which hopefully complements the previous presentations. And when talking about patient experience data, it's difficult to dissociate from the concept of patient engagement and incorporating patients and patients' views in all our work at the agency level. So in this respect, maybe just going back to the origins of the agency when it was created in 1995, we recall, and we will always put as an example, that immediately in the next year, we already had to respond to the calls of what at the moment we call activists, more than patient representatives, who wanted to really be involved directly in our discussions. And this was a step that we took with a lot of caution and a lot of concerns from many, but which resulted in very positive results and was perhaps the step which allowed us to build the system that we have today in place. A few years after, patients driven by legislation became members of committees at EMA with decision-making capacity as any other member. Then we established a framework to formalize this interaction and to really acknowledge the contribution that patients give. We created a formal working party, a standard working party, which still is functioning today. We later on created a specific function, acknowledging the need to really have dedicated staff to facilitate the involvement of patients. And then we explore new techniques, new methodologies like public hearings, ensuring that not only patients, but young people, carers are taken into account. Then we went through COVID and realized how critical is have patients and stakeholders involved in crisis management, and we actually put them in the core of our discussions in the emergency task force, in all our discussions on COVID. And I think more and more what we realize is that despite these efforts, we are still not there yet. We need to push more and to progress our strategies to ensure that in all the applications that we receive for new medicines, we have patient experience data supporting these applications. And as this data is really informing us what is really relevant, what is really important for patients, and that this information is taken when decisions are made. So just to illustrate where, because it's the question that we receive very often, where do you want to involve patients? I think the answer is everywhere. What we have realized is that it's a matter of resources, it's a matter of making priorities, but they really make a difference, whether it's during earlier stages of development, for example, during orphan designation, but during scientific advice, or even during pediatric investigation plans, or whether it's during the formal evaluation for approval, or afterwards during pharmacovigilance and risk minimization activities. And we can give a lot of examples. So this is just maybe to illustrate about scientific advice. We made a more in-depth analysis of the potential added value, the added value that patients have there. They're involved as experts. And you know that they inform, they participate in discussions about endpoint selections, of course, quality of life, feasibility of a study, any other aspects. And we cannot expect that the patient's contribution is going to make a change in the scientific advice given. Sometimes it's just endorsing, it's sometimes validating what is provided. But having them there is always of an added value, even if in some cases we can say that some of the recommendations made to the developer were modified because the patients gave maybe an insight which changed. So it's an example just to illustrate it. We have many more, but again, we should not be asking patients more than we ask to the experts. I think having them there already improves the outcome. It provides more trust, but I think ensures that we are not missing important elements that I don't think so regulators or developers can by themselves provide. So during the evaluation, more at the level of what we call the CHMP here, we don't have a legal basis to have patients members of the CHMP. However, we have developed ways by which they can provide input. And this is done, for example, systematically, they participate in what we call scientific advisory groups. We also involve them in oral explanations. Sometimes when there are other expert groups as well, they participate as experts. And more lately, we have developed what we call early engagement, which is that we contact specific organizations in the field of the new medicines which come for evaluation. And we ask them to provide information, or at least to give the opportunity to these patient organizations to give us information on daily impacts, treatment options, what are their perspectives, their perceptions, desired benefits, preferences. We do that at the beginning of the procedure so that it can feed and it can inform the rapporteurs who are leading on the evaluation. However, again, there is a need for a more systematic approach. There is a need for a more systematic approach to generate this data, and this is the initiative that we are trying to move on to define, to try to promote and to progress on the generation of patient experience data more systematically. We don't have EU guidance, and I think this has been highlighted in the previous presentation that maybe it would be good to have guidance at EU level. We had a recent workshops, and we came with the proposed definition that what we call patient experience data is data which is reported directly by patients or their carers without interpretation of a clinician, as has been highlighted before, and that in a way it collects patients' experience on their health status, symptoms, disease course, treatment preferences, quality of life, and the impact on the healthcare. Again, this is a draft definition. We hope to be able to agree with all stakeholders as part of the reflection paper that we are working. that I will mention now later. But patient experience data is a broader concept which covers patient-reported outcomes, other elements of quality of life, patient preferences, but any information which is generated again by patients without interpretation of a clinician. So it was also highlighted that we have to distinguish between the concept of patient experience data and patient experience evidence, which is data which is qualified and is valid for the scientific and to be used as scientific evidence as part of the assessment. Trying to explain why is important, I think maybe is a bit redundant. I think we all support that a lot has changed, a lot has moved, and I think we all agree that patients are the users of the medicines and they are the ones who know more. They are the real experts on the disease environment and their treatments. And we will have more patient-relevant outcomes if we do so. So they are instrumental in order to optimize the development and even decisions. So patient experience data will therefore be generated early during the development and will inform clinical trial design, benefit risk assessment, and post-marketing. But I think we have to go beyond that because this data also will inform cost-effectiveness decisions and reimbursement decisions, but also will inform clinical decisions at a later stage. So at EU level, again, this was highlighted as a priority in our strategies, in the network strategy, also in the regulatory science strategy. And I think we have to acknowledge that there has been a lot of progress and you know, EORTC is an example of how there is a huge push and a lot of research and trying to progress on this. However, systematically, it's not happening yet. It's not happening yet in all medicines that we receive. And I think there is a good moment now because it's acknowledged, but not also that there is a lot of opportunities in terms of digitalization, how data is being generated now, also in the context of real-world evidence, by which I think we need to progress and ensure that this data is more systematically introduced. Therefore, I think we have set up some kind of an action plan, and one of the key actions that we want to do is to develop what we have been asked by stakeholders, to have some kind of framework, a regulatory framework or reference on the best EU approach to generate, collect, and analyze data, patient experience data. So we are working on this reflection paper that hopefully we can release for consultation this year. And then another aspect, which also I think it will follow on the presentations before, is that we need to improve transparency as well. And we acknowledge that. We need to give clarity about how the data which is submitted is used by regulators and how it contributes not only to the outcome and the final decision, but also to even to the product information, if it is the case or not. And maybe perhaps there is some room for improvement here that we do it in a more systematic and transparent manner. So again, we have developed last year, we create an expert group with experts from the different committees from the network, from the different NCAs. And we are working on this reflection paper, which we hope to release for consultation later this year. So what elements will cover this reflection paper? Because it will not provide methodological guidance. I think we want to set up the principles and the framework. I think we want to have a clear message that EU regulators want patient experience data in their applications. However, the value of patient experience data maybe needs more reflection. And I think that's something which is where many of us struggle to try to define which is this value. And hopefully we can identify in this reflection paper, together with a proper definition, try to identify the limitations, the challenges, also elements of transparency, etc. We will be working on this reflection paper in parallel to other initiatives at global level, in particular with ICH guidelines on PED that you are aware of. And I think the idea is that we complement. I think that's why we say that in this reflection paper, we will not cover a specific methodological guidance because this will be covered more at the level of ICH. And we should ensure that we remain complementary and that we don't duplicate. And finally, maybe just to mention a key message that so far we would like to give in this reflection paper as well, and from EU, is that guidelines are important. Guidelines is relevant in order to progress on science, but I think we advise very much all developers to come early for scientific advice and discuss the need and the best way to generate patient experience data at the level of scientific advice. And this can be done individually for every medicine and on a case-by-case basis. And this will be perhaps the best way to progress specifically and maybe address the issues which were presented in the previous presentation. Also, we are more than welcome to look for qualification of novel methodologies and giving opinions, for example, on the use of novel PROs, etc. I think this is the message that we would like to give, is that it should be even ideal that systematically this is covered in every scientific advice that we provide. So I think I will stop here. I think I wanted to conclude how important for EU regulator is the value of patient views, the patient input in medicines. And I think it has to be at all levels in terms of engagement, but also in terms of the data which is generated. We have to acknowledge that PED is a new scientific discipline, and I think we need to collaborate a lot with all stakeholders in order to set the standards and to set them right in the way which can really help and foster the generation of use for future medicines. And these are the key to deliverables we will be working, and hopefully that we count on everybody's collaboration in order to progress on this. Thank you very much.
Thank you very much, Juan. So I think you clearly highlighted how important patient engagement is to EMA, and I think that this is a good transition to the next and the first session, which is addressing the question: how can PROs and health-related quality of life data inform regulatory decision as well as cover HTA needs? And this session will be chaired by Jaap Heinveld and Pierre Demolis. So Jaap and Pierre, this is now over to you. Thank you.
Thanks a lot, Caroline. Our pleasure. So let me introduce the first speaker of that session, Carla Torre from the Portuguese Medicine Agency. She's a member of the CHMP as a co-opted member and a member of the PRAC. So she's on the two aspects of benefit risk assessment on the safety side and on the benefit risk side. So who else could better present that topic? Thanks, Carla, for having accepted.
Thank you. Thank you very much. Good afternoon, everyone. Many thanks for the invitation to be here in this, I think, very relevant workshop. And my topic will somehow reflect some learnings from a recent review of patient-reported outcomes used on oncology trials between 2017 and 2020. This is the slide of my disclaimer. I declare that I have no conflict of interest. However, this presentation will reflect somehow my personal views and not the EMA or any of its committees that I am affiliated with. And actually, this was an academic research that was conducted at my University of Lisbon. And I see that we have Fabio in the room, who collaborated with the wonderful researcher and collaborated in this study as well. So just a brief flavor of the background, but I will, for the sake of time, I will skip some of this because I think it's very well known given the audience that we have. But capturing, as we all know, capturing patient perspectives during trials in oncology setting is a unique opportunity to collect information on patient experience of the disease, including its treatment, the impact of the quality of life. And here the quality of life is somehow more relevant than longevity. And also, as well acknowledged, cancer diseases, their respective treatment regimens are associated with significant negative symptoms. This was very, I would say, highlighted in the previous presentations as well, side effects and functional limitations. And because of that, over the years, these patient-reported outcomes in clinical trials gains special interest and is recommended by both regulatory authorities, namely the EMA and the FDA. So we decided in this scope, we decided to perform a study that aimed to analyze the data based on patient-reported outcomes on new oncology indications that were granted a marketing authorization by the European Commission between this time, 2017 and 2020, and also to identify the patient-reported outcomes related label claims that were granted. So in terms of the methods that we used, two independent investigators identify all the new oncology indications, including the line extensions that were granted a marketing authorization over the study period. And for each medicine indication, the EPAR, which is the European Public Assessment Report, and the summary of the product characteristics were extracted and retrieved. So in terms of the information that we extracted from the EPAR, we collected data on a therapeutic indication, on the study design, on the number of patients. And here you have in bold the data that we retrieved regarding the patient-reported outcomes, yes or no, and what measure was used, the patient-reported outcome endpoint as a primary, secondary, or exploratory. And we also, and I think from our view, this is one of the most important key of this study, is that we review all the assessors' comments regarding these patient-reported outcomes in what concerns to the SMP label claims. If there was somehow a reason behind the inclusion or exclusion of these claims. And of course, regarding the SMPC, we extracted data from the 5.1. Okay, so over this study period, a total of 128 oncology indications were identified that corresponded to 76 different medicines. And sorry for the small numbers in the slide. And over of these 128 indications that were approved, 100 included a patient-reported outcome measure in the confirmatory studies, of which the majority, roughly 72%, corresponded to solid tumor indications. And we also gathered information that over these 100 oncology indications, 37 was supported by double-blind RCT, 63 was supported by open-label trials, and out of these confirmatory trials, patient-reported outcomes was defined as the secondary endpoint in the majority of the studies that were reviewed. So overall, we found out that a total of 54 different measures were used. Of those 76%, the large majority were disease-specific measures. More than 80% of the trials used more than one patient-reported outcome measures, and a total of 240 patient-reported outcome measures were used across 100 indications where patient-reported data was used. But more or less 50 patient-reported outcome measures was seldom used in less than 10 studies. Overall, patient-reported assessments relied most frequently on EORTC and FACIT, which is very familiar for this audience, are both disease-specific measures, but also on EQ5D, which is a generic instrument. Actually, the EQ5D were used in approximately 2/3 of the studies, highlighting also this use regarding to HTA purposes. And regarding the cancer, we found out that the non-small cell lung cancer and the breast cancer were the cancer types with more medicines, including patient-reported outcomes measure in the confirmatory trials. And what about the SMPC claims and the EPA reviewers' comments? Well, our study showed that 22 indications, so roughly 70% of the indications that were reviewed included patient-reported outcome labels in the SMPC. Half of these was supported by randomized open-label studies and 45% by double-blind RCTs. Patient-reported outcome was selected as a secondary endpoint in 16 studies, roughly 72%, and as exploratory in 4 studies. And now the part of where we extracted the EMA reviewers' comments, when I think this is the beautiful part of this study, because it somehow helps us to pave the future. So we found this for 76 indications had EMA reviewers, namely CHMP-assessors, comments on the patient-reported outcomes in the EPAR. And for 34 indications, reasons for not labeling or for not including patient-reported outcome in the SMPC were provided. But we also have, at least for 2 medicines, if I recall correctly, the other side of the coin, where the dossiers didn't have patient-reported outcomes, but the assessors mentioned that it would be important for the benefit-risk, for supporting the benefit-risk decision and for the benefits of patients that the marketing authorization holder that could somehow collect this important data. So we do have the other side of the coin as well. So in terms of the comments that were raised by the CHMP assessors, so somehow we can group them in 4 main blocks, I would say. So we have one block that is related to the study conduct, another one to the patient-reported outcome selection, the third one related to missing data, and lastly, the study design. But of course, that we could somehow, given because it's not an open-ended question, some of the comments could somehow have one or more one of these points. And we can see here that the most recurrent argument pertain actually to the caution needed for patient-reported outcome data interpretation when using an open-label design, followed by the clinical relevance. So it was hard to interpret this data, and how this data could be used was somehow unclear, limited, and missing data. So these were the most frequent reasons gave by the CHMP assessors for not included the patient-reported outcomes in the label. So as in any other study, we do have strengths and limitations. I will start for the limitations. So this review examined only the new oncology indications that were approved by the European Commission between 2017 and 2020. Hence, we cannot say that this European patient-reported landscape is fully characterized, because this study was only for a limited study period. And we, as a research team, we inferred that when including a patient-reported outcome in a clinical trial, a patient-reported label was intended, which could have biased our result interpretation, and as we know, could not be the main intent for the company to be included as a label as well. And our review was based only on public available information. So we didn't include the assessment reports And we have much more information in the assessment reports, but they were our study reviewed only the EPARs. And one of the major advantages I have already highlighted is related to the extraction of the reasons for not including these patient-reported outcomes in the label as compared to other studies. So in conclusion, despite growing recognition on the value of patient-reported data, patient-reported implementation remains a challenge. This is not new. Between these study periods, EMA granted patient-reported labeling to 22 out of 128 oncology indications. And we found out that roughly 78%, including these patient-reported outcome data in confirmatory trials, as compared to a period previous for the conducting of this study, we found out the higher patient-reported outcome used, but lower patient-reported outcome labeling. But I don't like to compare. Perhaps sometimes it's not fair because the therapeutic landscape evolves over time, and we might comparing orange with apples. But the main message is that assessors highlighted several key concerns were identified regarding patient-reported implementation that including the rationale, the study conduct in what it comes to data collection, training, management, analysis, influence of the study design, missing data, and the patient-reported outcome measure selection. So some summary remarks that might be useful for the next discussion panel, I would say. While patient-reported outcome implementation remains challenging, there is undoubtedly added value benefits in the use for research, for clinical practice, for HTA decisions, etc. But methodology, robustness, and consistency of the outcome reporting are key. A lot of effort should be put on protocols, on planning, on mitigation bias strategy, and also, and it was mentioned by Juan, on early dialogue with regulators is very, very important. Otherwise, it will be a waste of resources and ultimately it won't benefit the patients, which is our main purpose. Thank you very much.
So many points to be discussed with the panel. Thanks. Nice introduction to the panel discussion. The next speaker is Vishal Bhatnagar from the FDA. I hope I haven't pronounced too wrongly your name. You are participating remotely, so welcome and thanks a lot for your participation.
Thank you so much. So my name is Vishal Bhatnagar. I'm a medical oncologist and associate director for patient outcomes in the Oncology Center of Excellence. I'm pleased to be part of this important workshop, and I wish I could have been there in person with you all. Next slide. I have no disclosures, and as previous speakers have mentioned, this is my personal opinion, and the contents of this presentation are current thinking and subject to modification in the future. Next slide. So all too often, patient-reported outcomes are included only in late-stage or registrational oncology trials. We know that it's feasible and actually advantageous to include patient-reported outcomes during dose optimization, as well as late-stage trials in oncology drug development. It's important to measure the patient experience all along the drug development continuum, not just for labeling, but in order to inform safe and tolerable medications for oncology. This measurement may look different in early phase studies compared to late phase studies, but asking patients about their symptoms and function can be done across the drug development continuum. Next slide. Taking a step back, at FDA, we have a dedicated set of resources to further patient-focused drug development. We know that there are systemic ways of gathering the patient perspective in oncology, And much of this is driven by the negotiated user fee authorizations over the past few years. Also, US legislation entitled the 21st Century Cures Act also highlights advancing patient-focused drug development in the form of a 4-part guidance series. FDA receives a wide range of types of marketing applications. On the left, you'll see common clinician-reported data that's collected in almost every trial. That includes measures like overall survival, clinician-reported safety, and then health utilization data such as hospitalizations. On the right, you'll see patient-generated data, and to create some consistency in the field, OCE has designated a core outcome set that should be collected in every cancer trial. This includes disease symptoms, symptomatic adverse events, overall side effect bother, physical and role function. And this isn't meant to say that these are the only patient-generated elements that should be collected, but this represents the bare minimum of patient-reported outcomes to collect. Next slide. An important consideration for patient-reported outcome assessment is the frequency of assessment. As you can see on the left are those core patient-reported outcomes that I mentioned previously. And then in terms of assessment frequency, this really boils down to frequent assessment at the outset of the trial when patients are most likely to experience side effects from treatment or changes in function. As the trial continues, the assessment timing spaces out to monthly and then every few months to detect long-term changes, but these time points should be carefully considered based on disease and treatments being studied. Next slide. So it's not just sufficient to collect this data and then do nothing with it. It's important to pre-specify PRO objectives and endpoints. So for example, PRO endpoints can describe the patient experience while on treatment, inform safety and tolerability, or inform efficacy. So for regulatory purposes, you need to ask yourself, how would you like this data to support your product? Are you submitting the data as purely supportive exploratory data to inform benefit risk, aka totality of data? Or are you interested in incorporating descriptive safety and tolerability data in an FDA label? Or are you intending to provide substantial evidence of a treatment benefit, such as drug A improves disease-related pain? Based on the PRO objective, there needs to be careful consideration of how to craft that PRO endpoint of interest. Next slide. As an example, we've included important patient-reported information about tolerability in Section 6 of labeling. In this case, we included patient-reported visual disturbance, which was important to communicate to patients. Although clinician-reported visual changes are included in the label, that information only includes the incidence and severity. In this trial, patients were asked using the VSAC-ALK about their experience regarding visual disturbance, and this provided valuable tolerability information. Specifically, the patient-reported information includes timing, duration, frequency, and impact on activities. I've personally heard from patients that this information is reassuring and valuable and is shared among lung cancer patients initiating therapy with this drug. Next slide. Patient-reported treatment-related symptoms can provide valuable complementary information, just as I mentioned in the last example. And this information may not easily be gleaned purely from clinician-reported safety data. Unlike how symptoms are typically assessed in an office by a clinician, patient-reported outcomes, in this case PRO-CT-CAE, asks patients the same question on a regular basis every time, and patients will score their response using the same criteria each time. So on this slide, we have an example of diarrhea. For standard safety assessment in a trial, a patient may come to their clinic visit and the clinician may ask a broad question, how are you feeling? And then the patient may offer that they've been having diarrhea. The clinician then may quantify the grade of that diarrhea through both the frequency and severity and whether medical intervention is required. But the patient-generated item, the PRO-CTCAE question here, is often assessed electronically, perhaps even from the comfort of a patient's home, directly asking the patient in the last 7 days, how often have you had loose or watery stools? And then the patient will fill out for themselves the frequency of the diarrhea using a standard categorical response. And as you can see, I think the most important take-home point here is that the CTCAE grade of 1 or 2 encompasses an extremely wide range of experiences. And the use of a PRO can provide more granularity behind a grade 1 or grade 2 CTCAE event. Next slide. In addition to specific symptoms, another core PRO outcome is overall side effect bother or impact. This is a way to capture the totality of side effects, as multiple grade 1 or grade 2 CTCAE events in combination can cause substantial tolerability issues for patients, perhaps even precluding administration of the medication. These are simple questions, easy to integrate into clinical trials, and FDA has funded research into how best to analyze those data. The NCI in the United States is also doing work with the summary measure as part of their tolerability consortium. Next slide. So much of what I've stated in this presentation, including the core PRO concepts and example assessment frequency, as well as other important regulatory considerations, are included in a guidance, a draft guidance by the Oncology Center of Excellence, and this was released in 2021. Next slide. Specifically to dose optimization and tolerability assessment, the Friends of Cancer Research assembled a group of stakeholders and published more details in this white paper, which was released in 2022. Myself, Paul Klutz, who you will hear from later, and other US FDA colleagues were involved in the development of this white paper, and it includes important information that can guide early phase development. Next slide. So in summary, clinical outcomes assessments can complement standard efficacy and safety measures. PRO is one type of clinical outcome that can do this. These PRO concepts should be well understood, and instruments should be fit for purpose and well defined. Tolerability can and should be assessed in all oncology clinical trials, including dose escalation and expansion trials. Item libraries are a great tool, and I did not mention that specifically in my presentation, but I think it's been mentioned already and will be a topic of discussion later today. And item libraries can be used to meet needs of more than one stakeholder, not just US FDA, but other regulatory agencies, HTAs, and all academics as well. Well-collected and meaningful PRO information should be communicated to patients, caregivers, and providers. And as we heard in the previous presentation, there is room for more and better incorporation of PROs into labeling. And I think that by use of the methods that I just mentioned, perhaps we can improve the chances that PRO can be included in labels in a thoughtful and clinically meaningful way. Next slide. So thank you to all those that I work with at the US FDA, and most importantly, the patients and advocates who participate in our initiatives. Thank you.
Thank you very much. Now it's my pleasure to introduce the next speaker to you, which is Dr. Beate Wieseler. And she's the head of the Department for Drug Assessment from IQWiG. And now that stands for Institut für Qualität und Wirtschaftlichkeit im Gesundheitswesen. Dankeschön. Based in Cologne, I think, Köln. Okay, the floor is yours.
Thank you very much for this nice introduction, and sorry for the difficult name. Thank you very much also indeed for the invitation to this important workshop and the opportunity to discuss specifically health technology assessment requirements, because actually patient-reported outcomes and including quality of life are indeed very important in our assessment framework, and therefore it's great to be here. These are the topics I would like to discuss with you today. First, I will provide you with a brief overview of the decision-making frameworks that are based on health technology assessments. And I would specifically address the use of PROs, including health-related quality of life in health technology assessment. I will describe a few general challenges we are seeing with PROs, and I think this will very well resonate to Carla's findings from her study. And I would like to briefly address two specific issues from an HTA perspective that are currently under discussion, that is post-progression PRO data collection and item lists. So if we look at decisions that are supported by health technology assessment, HTA, then we have two main questions. These decision-making frameworks are mainly used after drug approval by regulatory agencies. And then that means new treatments become available to clinicians and patients, but they are not in isolation. They meet a treatment landscape, and then of course the immediate question is which would be the best treatment in a situation for a specific patient. And therefore what we are doing is to compare new drugs with the available treatment to be able to inform the choice of the best treatment for patients. Another important question we are addressing is we want to describe the value of a given treatment compared to what is available, and this is meant to enable pricing, because in Europe we are aiming at universal health care, and this is only possible if we can keep our healthcare system sustainable. Therefore, the price of a drug based on its value always is an important means to ensure the sustainability of our healthcare systems. The data we are using to answer these questions are generally comparative effectiveness and safety data, and sorry for the typo, versus a standard of care. So as I said, we don't look at a new drug in isolation, but we always compare to what is available and what is considered the standard of care in a given situation. And then we are making decisions, and there are different types of decisions actually that are supported by HTA, and this partly depends on differences among European countries, which are again due to differences in healthcare systems and decision-making in healthcare systems. So basically you could distinguish between two types of decisions. So there are countries which decide on reimbursement and pricing based on the clinical added benefit, and clinical added benefit would also include less harms. And there are other countries which decide on reimbursement and pricing based on cost effectiveness. That is the price of a drug given a certain benefit. And of course, all these things are connected. So obviously, the cost effectiveness is also informed by the clinical benefit. But there is also information that directly goes into the cost effectiveness, and that, for example, are utilities which are based also on patient-reported outcomes. And then these decisions on the question, is there a clinical added benefit or is this drug cost effective, is used to support individual treatment decisions, is used to support clinical guideline development, to again support treatment decisions on a system level, and is also used for pricing negotiations and pricing decisions and reimbursement decisions, which then of course also have an impact on availability of treatment and therefore again on treatment decisions. As I've said, the healthcare systems within Europe are partly different, and therefore the decision-making frameworks are different. However, across European countries, HTA bodies agree on what they want to know, on the important types of outcomes they want to know. And that is mortality, morbidity, and health-related quality of life. So overall, this is describing, and that I think actually is a very old regulatory definition. And we want to know how a patient feels, functions, or survives. We could differentiate that a little bit further. So morbidity would cover symptoms, complications, and function. And health-related quality of life is defined as the impact of the disease or its treatments on these three dimensions: the physical, emotional, and social well-being. And if we look at this portfolio of endpoints, we see that actually most of that is best collected from patients directly. So patient-reported outcomes have a very important, play a very important role in our decision frameworks, in our assessments. Concerning morbidity, symptoms can obviously only be collected by patients. Also, many complications are reported by patients. Function is sometimes assessed by a clinical assessment, but very often also is a patient-reported outcome. And the whole area of health-related quality of life is just by definition a patient-reported outcome. So I think this picture very well shows how important patient-reported outcomes are for our assessments and our decision-making, and therefore patient-reported outcomes have played a role in health technology assessments for years now. If we look at this decision framework again, so we see that from these comparative studies, results from patient-reported outcome go into answer the question, is there a clinical added benefit of this new drug as compared to what is available? Patient-reported outcomes inform cost effectiveness by utilities. And then, of course, we also consider this important because we want to answer the questions patients are having when they make treatment decisions. And I think that was also described earlier, that if patients have the choice between two treatments, they want to understand what that means for their life and what the alternative option would result in. So that is one use of this information. And then these data are also criteria for pricing decisions. For example, in Germany, by law, the price of a new drug is influenced by the health-related quality of life compared to the standard of care. So given this is so important, this is just a summary of current challenges with PROs. And I think the overarching issue is that the methods used for planning PRO collection in studies, for analyzing this data, and for reporting PROs are often less robust than for other endpoints. We have insufficient pre-specification of PRO data collection and analysis. So we still very often see PROs as exploratory endpoints without any pre-specification. We often see it as secondary endpoints, but without proper pre-specification. And we have seen, I think, in the earlier example, to what extent this would need to be pre-specified to be really robust and not data-driven in the end. And we still often see that this data isn't even reported in the main clinical study report, but provided like as some PRO report just meant for cost effectiveness or something like that. So we really would see that obviously in the main clinical study report. Then there are issues with the interpretation of PRO results, because you might end up with kind of abstract differences in points on a questionnaire that is difficult to understand. What are relevant changes? What are relevant treatment group differences? But there is ongoing work on that also in the EORTC consortium. The problem of missing data has been raised. This is mainly due to insufficient follow-up with data collection. We at the same time see studies that have a very, very high compliance with data collection. I remember one very impressive study from EORTC, actually, which I think was a prostate cancer study over 10 years, which had 80% compliance with PRO collection at year 10, and that was very impressive. But again shows that this is possible. At the same time, doing this right is not rocket science. So in principle, we know how to do that with maybe a few specific issues like the interpretation or some specific analytical problems. But it's not that we can't do it. I think we just have to understand the value of these endpoints to improve the data collection. And because we consider PROs such important endpoints, we would like to see a robust methodology so that this data can indeed have an impact on our decisions. Maybe very briefly on these two specific issues. We have a problem with the current standard of oncology study designs. The current standard is that PRO data collection stops with disease progression. And this is not answering our questions. I mean, patients are not interested in the quality of life up to disease progression. They are interested in their quality of life for the rest of their life, even after change of therapy. So that isn't answering the patient's question. It's also from if we go into cost effectiveness analysis and we're using quality adjusted life years, what is happening now is that we are guessing how the quality of life after progression is. We are modeling some data rather than just collecting them. And of course, we are aware that it might be a certain challenge to continue this data collection for very long, but I mean, I think we could find some intermediate solutions that would be very helpful. But indeed, we currently see as a standard that PRO data collection is stopped at progression, which leads to an additional, maybe more methodological problem that if the progression differs between the treatment group, we also have an issue with the analysis of these different observational periods. A few words on item lists. We are fully aware that item lists may be a possibility to optimize data collection. We also are fully aware of CAT instruments and the advantages that this has. I think we all agree that also with item lists, we need robust methods to develop these lists, because we don't want to go into selective compilation of item lists to kind of optimize the outcome for a certain treatment in a specific trial. That is what we obviously want to avoid. We also still want, if we have an item list, cover the complete construct we are looking at or the complete symptoms of interest. But I think health technology assessment has a specific additional issue, and that is coming from the fact that we are always looking for comparative effects. So we are always looking for the new drug versus the standard of care. So any item list optimized for the new drug would be an issue, would not result in a fair comparison. So we at minimum would need to consider both the new drug and the comparator. We are often also going into indirect comparison, that is using data from different studies. If we would use different item lists in the different studies, we can do our assessment. So I think overall, we are a bit nervous about that. So these standardized questionnaires allow, for example, for indirect comparisons because we have the same data set. So this is just a caveat that if we end up in sets of studies with isolated different item lists, these are less relevant for us. And this just as a point on our wish list. And so maybe as a conclusion, PROs, including health-related quality of life, are important for health technology assessment. Actually, they are as important as any other endpoint. And therefore, we need high quality data on this. And we consider this very important because this data represents the patient's voice directly. It directly provides a patient's voice to our decision making. And because this data is so important, we need robust methods for study planning, data collection, analysis, and reporting and interpretation. Thank you very much.
Thank you very much. Couldn't agree more with many of your observations and your worries, I think. Maybe it's interesting to mention in this perspective that there's also a utility variant of the C30, which we developed, the QoL C10D. So through the questions of the C30, you can also calculate utilities. But let's move on to the next speaker, which is Douwe Posmus, who's from the University of Groningen. And I challenge everyone who is not Dutch to pronounce that word in a proper way. And he's also the seconded national expert for EMA for the Netherlands.
Yeah, thanks, Jaap. So you can always just, refer to the second title, seconded national expert, so then you don't have to mention Groningen. Anyway, I'm going to give, let's say, a slightly more, I would say, explorative presentation, in the sense that we have been thinking about ways to potentially include PRO data into the benefit risk assessment. So this is not at all something that is actual current practice, but it is more some ideas that we were having. So as you all know, when we are looking at the benefit risk assessment currently in oncology, it's mainly about trade-offs between overall survival or progression-free survival and toxicity, where toxicity is typically clinician-assessed toxicity, usually grade 3/4 events that are then being compared against the increase in overall survival and progression-free survival. So we have an example on the slide. A typical improvement could be a 12 months improvement in median PFS with the experimental treatment compared to 9 months with the control treatment. You also see an increase in grade 3/4 toxicity incidence. And the question is, are you willing to accept this increase in toxicity risk for the observed benefit in terms of PFS? And while these are obviously very important or key benefit risk trade-offs, they do not always capture the full picture, as was already being alluded to by previous speakers. Although grade 3/4 toxicity captures the severity, it doesn't really capture the frequency of the event. Right? So severe event that lasts for 1 week is kind of counted the same as an event that may go on for, let's say, a few months. And also an event could be recurrent, which is then not really reflected in the toxicity data. So instead of making trade-offs between these incidence rates, another possibility could be to focus much more on, let's say, the patient health state, a bit comparable to how HTA would be performing these assessments. But then by trying to capture the patient's health in terms of PROs. And then you could be looking at trade-offs like expressed in the example. In the final bullet is 9 months progression-free survival with toxicity, followed by 2 additional months progression-free without any toxicity. Is that now preferred over a situation where you have 9 months PSFs without experiencing any significant toxicity? So you see, you're still making trade-offs between toxicity and progression-free survival, but you focus a bit more on the time spent during the progression-free state. So what we wanted to do is we wanted to get some better understanding of well, how these two different types of benefit risk assessment would look like. And for that, we used a hypothetical case study. So this is simulated data based on a well, previous trial that was being performed by EORTC. We have some data we received from Cornell. And based on that, we did some simulation. And for the simulation, we focused on 4 main outcomes. Time to the onset of systematic adverse events, collected using a PRO, the duration of the toxicity event, and the traditional time to disease progression and time to death outcomes. And in here, this is simulated data, but you can imagine that you have a PRO collecting symptomatic adverse events. And based on the patient classification into the severity, you can either say this is a severe or significant event, or this is a slightly non-significant event. So based on that information, you can kind of dichotomize whether the event is severe or not. And we also simulated the duration. And what we then wanted to do is take the simulated data and then perform a more traditional benefit risk analysis and compare this to an approach where we would be focusing more on the amount of time spent in the state. So if we then start with the more traditional analysis, then we have the 2 Kaplan-Meier survival curves, and we have some information about the toxicity incidence. So if you look at the survival curves, we have the progression-free survival on the left, and the overall survival on the right. And then we see that there is no real difference in terms of overall survival. So the efficacy is mainly in terms of prolonging time to progression. And it comes at the price of an increased incidence in toxicity. So if you look at the benefits, then we see we have roughly a 3 months increase in median PFS against a more than 30% increase in the proportion of patients experiencing severe toxicity. Right, and then in the traditional benefit risk assessment, it is really about what is the maximum acceptable risk we are willing to accept for this 3 months increase in progression-free survival. Now, depending on the impact and duration of toxicity, this may not tell the complete story. So what we wanted to do next is estimate the time spent in a certain toxicity state. And for this, we used a statistical approach called multi-state modeling. For now, the underlying statistical technique is not that important, but the idea is that you essentially divide the states that the patient can be in over time. And in the model, we make a distinction between 4 health states: progression-free without any significant toxicity, progression-free with significant toxicity. Then we can have progressed disease, and in the end, the patient will make a transition to the death state. And you see that whilst being on treatment without progression, patients can go back and forth between, let's say, episodes with more and less toxicity. Right, so by fitting this model to the simulated data, we were kind of able to estimate the time spent in each state. And then you get these, well, I would say nicely looking figures, but let me try to explain them as well, because it may be a bit complicated the first time you see them. So what are these curves displaying? They are displaying the prevalence or the proportion of patients in each of the 4 states over time. Right, so if you would have long enough follow-up time, eventually everybody would die. So in the end, everybody is in the death state. In the beginning, everybody is still progression-free and there is no toxicity. And then you can see nicely how the treatment develops over time. Now, when you look at these 2 figures, what you kind of hope to see ideally is a situation where you improve overall survival. But in this particular case, as we have also seen in the traditional analysis, overall survival was not significant. So we do not see a direct, a huge distinction in the red area. So the beneficial effect of the treatment is mainly in terms of extending progression-free survival, but it comes at a certain cost. And the cost is that you have more toxicity. So this green area is the best state, so being progression-free without any significant toxicity. And you see that is actually slightly higher in the control group compared to the experimental group. So on average, patients spend 6.4 months without any toxicity in the experimental group and 7 months in the control group. But patients in the experimental arm spend more time being progression-free, but with toxicity. In this case, it's roughly 4 months with the experimental arm, and severe toxicity doesn't really happen that much in the control arm. So now we have a slightly different perspective on the data. You see with the experimental treatment that you have a slight overall increase in progression-free survival, but a much larger part of the progression-free time is actually being spent with significant toxicity. So then the question is, are you willing to spend more time being progression-free, but with experiencing a significant toxicity compared to spending a slightly longer time in the progressed disease state? And this all depends on how you would value these health states. So a limitation of this approach, and it was mentioned by the previous speaker as well, is that currently we don't have any data on toxicity in the progressed disease state. So the distinction is currently a bit strange in the sense that we try to very carefully distinguish toxicity status in the progression-free state. But in here we need to currently come up with some kind of overall estimate of how severe this state is. So in an ideal situation, we would also have, let's say, quality of life or PRO measurements there, so that we can kind of also subdivide this state into, let's say, affected by toxicity or not, to get even a let's say, more comprehensive view of the benefit-risk profile. So just to summarize, so this is very much exploratory, but what I like about the approach is that it may complement these more traditional benefit-risk assessments. And it offers a fairly natural way to take into account data collected with PRO measurement. And I didn't show that for now, but you could also go one step further. And you could, let's say, assign formal utility weights to these states. And then you can perform a quality-adjusted survival calculation comparable to quality-adjusted life year calculations in HTA, but then maybe with values directly derived from patient preference studies using these outcomes. So some next steps, well, like I said, this is very much exploratory, but is it actually useful? Is there value in focusing on comparing time in a certain state compared to looking at traditional cumulative incidences? Well, that's all I had to say. Thank you for your attention.
Thank you very much, Douwe. And to further illustrate, both you and Francesco will probably attend part of the next quality of life group meeting in the end of this month. So that's already an extension of the collaboration, I would say. We're perfectly on time. I think it's time for the coffee break, and let's gather around 3:30.
Good afternoon. Welcome back, everyone. I'm very pleased to chair this part of the session. My name is Peter Mol. I'm a professor of drug regulatory science at the University Medical Center Groningen, so I can pronounce it as well. And I'm the current member for the Netherlands in the CHMP. Have done a lot of scientific advice work. Very interested in innovations in the way how we regulate medicines. But enough about me. I think together with Jaap Reineveld, who has introduced himself before, we will try and chair a panel discussion with no less than 9 participants. We rather do than introduce everyone individually. We will go along the line of the people here on the screen and ask them to present themselves with especially a short reflection on what they feel in the setting of the PRO is really key to their use of it. So what do they believe? How can they make better use of PROs from their perspective? And that would be a very brief introduction. So please stick to about 2 to 3 minutes in that presentation about yourself and your view. And then we will come to the topics that we want to discuss here more on what is needed for a more systematic use of PROs and submissions, differences among regions, what kind of standards, could there be harmonization, and what concrete next steps will we propose, as this would be the intent also that Jaap and his team would try and come to a sort of white paper based on the meeting on the messages that are received here. So that is where we wanted to go. And so without much further ado, I would like to ask Paul Klutz of the Klutz, I'm not completely sure, of the FDA to introduce himself and briefly his main take on what needs to be done for a PRO to be useful. Thank you.
Thank you very much. I'm going to try to pronounce my own name in a European way. I'm Paul Gustav Klutz. My dad was from Germany, and so I like how Jaap has been focusing on pronunciations. I'm really happy to be here. I've spent a lot of time looking at patient-reported outcomes for the FDA. I'm currently the Deputy Center Director for the Oncology Center of Excellence. Briefly, for me, I think I can sum up how I think the data can be used best for our purposes, which is to really try to isolate the effect of the drug on the patient and their disease, is more intentional thought behind how to incorporate the patient-reported outcome assessments within each clinical trial. And I know that there's been a lot discussed about some of the details about that, and particularly in the area of harmonization that might need to happen with different opinions on things like assessment post-progression or using larger validated instruments versus going with a more item selection from a library approach. So I'm looking forward to that discussion, and I'll wrap it up there so we can get through the rest of the panelists. Thank you.
Okay, thanks. So we would move on to Maxime Sasseville of Health Canada.
Hi, I'm Maxime Sasseville. I'm a manager of oncology division at Health Canada, and I've also been involved in a number of patient-reported outcomes initiatives over the recent years. I think where Health Canada is moving in terms of quality of life, I think in our experience, where we've seen the most recent progress is to start seeing sort of, we call them GEMS. I call them GEMS, like drug development programs that are with very robust patient-reported outcome collection, where I think the interest in having very robust statistical design has allowed the companies to come up with lots of attention to the details of their of how they collect and analyze their data, and I think that's where we see increasingly a number of good programs coming our way, and it makes our life in a sense more difficult, but to reject the patient-reported outcomes in our decision making, but also makes our life a lot easier because we find the attributes, the quality attributes that we want to see in robust quality of life data sets. So tools that are fit for purpose, tools that are age appropriate, robust thresholds. We've seen recently, for example, drug development programs that relied on tools that were used in clinical practice with vast publication records. Even though they were in very small, sometimes patient population, they still had very valuable insights into the disease state and how patients were faring on drugs, on the proposed drug products we could see. So I think the field overall is going in a good direction, and I think those gems are the ones that we should leverage to show that it's definitely feasible and within reach to make good use of quality of life data.
Okay, thanks very much, Maxime. The next panel members in the audience, fortunately, it's Friedrich Wittenberger from Swissmedic. The floor is yours.
Yes, thank you. Exactly. My name is Friedrich Wittenberger. I'm a clinical assessor in the oncology division at Swissmedic, which is the Swiss agency for therapeutic products. And well, I guess I could reiterate many of the points that were made already in the talks and by my predecessors. I think for us, it's very important. Oh, sorry. It's very important that there is actually a clear QoL hypothesis in the statistical plan in order to include it in the label in the end. So I think it's also something to do with our legislative surroundings, but I think that would be a point that is very important to us. And regarding a more comprehensive or a more harmonized methodology, a more robust methodology, I think what we would also appreciate is maybe a more common way of visualization of the data. I mean, with many time to event endpoints, for example, we are very used to Kaplan-Meier curves, and which gives us also a feel to compare data easily across trials, and to move towards something like that for quality of life data would be very helpful, I think.
Thanks very much. The next one is online again. He's from Japan, so it's a pretty long way to travel for one afternoon, I would say. Dr. Shun Tasuka. Tasuka, are you here?
Yeah, I can hear you. Thank you for your introduction.
Welcome. We can hear you as well. Would you like to introduce yourself?
Yes, I'm Shun Tezuka, medical officer in PMDA, regulatory in Japan. My specialty as MD is oncology, especially in hepatobiliary cancer, gastroenterology, and endoscopy. I'm very grateful to participate in this workshop. The presentation so far have been very informative. In Japan, there have been only one small guidance about PRO issued last year. Of course, we do recognize the importance of PRO and HRQoL, but having difficulty taking effective steps forward. In oncology, needless to say, efficacy endpoints such as OS, PFS, and objective response rate have been well established. At the extreme, in Japan, anti-cancer drug could be approved without a PRO endpoint. Of course, if PRO results are submitted, we evaluated the PRO result in the review process. However, since the evaluation method of PRO has not been established regarding anti-cancer drug, we currently do not include PRO result in assessment reports or labels in Japan. I must say Japan is lagging behind. I'd like to use this opportunity of this workshop to collaborate with you and move forward together. Thank you.
So thank you for that perspective, and I think it's really good to have here that discussion. And it's also a pleasure to introduce the next speaker, Harold Ensman, who is the chair of CHP and whose chair I'm currently holding warm, which is a good feeling. Harold, please.
Yeah, thank you for the introduction. My name is Harald Ensman, and indeed I have the honor to chair CHMP. That is the committee that in Europe will in the end give an opinion about a positive or negative benefit risk for medicine. Now, I would like to come back to the very, very beginning of this wonderful meeting when Pierre de Molis said very clearly, and I can only re-emphasize that, yes, we as regulators, we do look at the patient-reported outcome data we get, and we do consider those in the regulatory decisions. And allow me to remind everyone, including ourselves, that the regulatory decisions cannot be an end in their own. They can be only one step to the very important, ultimate important decision that in the end, the individual patients make against or for a specific treatment. And helping that, supporting that, we do want and we do need the patient-reported outcome data, and we try to make the best possible use. And very clearly, and again, something I need to remind everyone, this best possible use will depend on the scientific quality of this data. Now, best possible use does not imply here an all or nothing approach. It does not force those PRO measurements we receive into any kind of black and white scheme. Rather, what is important is that we make sure that the scientific quality is proportionate to the objective. Importantly, to do this, to make sure that the scientific quality is proportionate to the objective, the very objective must be known, should be very, very clear from the beginning. That means when designing a trial, as early as that, it should be clear to everyone if the PROs you will take will be the primary endpoint of this trial that defines in the end whether the study is successful or not, that is intended as a crucial basis even for the marketing authorization, or different but not less important, the data collected are meant to provide information for labeling, information that is intended to support the patients later at the most important final ultimate decision, the individual decision for or against a certain treatment. So bottom line, what I'm trying to say here, we need to target PRO measurements to a predefined objective. That is where I see quite a significant potential to diminish this difference we all perceive between the expectations we currently have in this data and the impacts they ultimately have on the decision making. So, summarize. Maybe it's time to, similar to the targeted therapy we've gotten so used to in oncology, we may want to start to strive for a more targeted PRO measurements. Thank you.
Thank you. Peter and I have one microphone, so we are not allowed to interrupt each other unless we grab the microphone. So we flip it every time. The next member of the panel is Anja. She's in the audience. Anja.
Yeah. Anja Schiel. I work at the Norwegian Medical Products Agency. I have to read that because we just changed our name. The abbreviation stayed, but you have more tasks with the same kind of people. Always a good thing to do. But obviously, I'm here today in my role as an HTA representative, but my title is Lead Methodologist in Regulatory and Pharmacoeconomic Statistics. So I'm obviously just using a fancy title for being a statistician, which is one way on looking on everything, including the PRO data. I do regulatory assessments, so I'm very familiar with the CHMP and scientific advice perspective, of which I'm also a member. And we do HTA assessments. Our assessments are slightly different from what Bettina presented. Beate, sorry, Bettina's sitting there. Yeah, sorry. What Beate presented and is more related to what Dao presented. Because what we do is we generate a hypothetical future with the information we are getting, which means that we love precision, but we can live without precision because we can model something. It just depends on our assumptions. PRO data for us are obviously very important because we are also, just like you heard from Maxime, for example, we are using the quality, and it says quality of the life in that term. So what we do is we need information that is very clear to us to inform our assumptions. And one of the problems that we see with the reporting of PROs, mostly, not necessarily the quality of the PROs, but the reporting of the PROs, is it the same I saw in the other dossier? Is it the same and just more fancy figures? Or is it something completely different? And that is often very difficult to derive from the information you get, because as long as we don't have consistent, systematic reporting of the essence of the tool, we spend lots of time on not assessing or interpreting the results, but rather trying to figure out if it is what we think it is. That's why I'm also involved in the CISACO, and I find it extremely important that we are going to reach some kind of standardization, because as an assessor in the CGP system, as a statistician, I just love the fact that I know exactly which module I'll have to open, go exactly to the protocol and the figures that I need, and get always consistently the same figures. This is what we need for PROs, because then we have time to actually do what we are supposed to do, assess the quality and the information in the PRO tools, instead of trying to figure out what we're really looking at.
Thank you, Anja. And then I think indeed we move to your neighbor, to Bettina Riel or Rule. I think also today it's really the practicing of German names here. But who's bringing in actually the patient perspective now to the patient-reported outcome. So, well, after 7 speakers, Bettina, go ahead, please.
Thank you. It's Röhl. I am not from Groningen, but I am German, but I live in Sweden. So I think I founded the Melanoma Patient Network Europe after my husband died of melanoma, and I'm scientifically trained, so I'm an MD and I have a PhD in molecular biology. And I ended up in something that is more, I think, closed, or that is qualitative science, because of my advocacy work. And my organization is one of the weekend founder organization that is the Working Group for European Cancer Patient Advocacy Network, which is quite a mouthful. So I think my perspective is a bit maybe complementary to what we have so far discussed. I think this has for me been a lot about internal validity. I'm more concerned about the external validity, because we should not forget, just because a patient reports it doesn't mean that it is relevant to the patient. We can ask people to report plenty of things and it can mean absolutely nothing. So one of the things I've been worrying about, and I had the opportunity to meet many of you in that function, because we were obviously not happy with what was asked. So I think that is one of the big challenges. I really think that like what Pierre gave as a setting at the beginning, to think about a disease in its stages and to think about different population moving through a disease pathway, is something else that I believe is very important. To understand the granularity in which, like, of a situation in which a decision is taking place, and how to understand what matters to people. So from my background as a physician, I believe our ambition should be to do right by the person, and instead of projecting our ideas or being too close to the method and missing the point of why we're doing this in the first place. I actually also think, and I've been on record for having said that before, that the EMA is always underselling what they do in terms of patient involvement, because something that I personally have found extremely valuable is this space of regulatory science, and the more, as Dauber already presented, conceptual and research-oriented frameworks, because I believe that this is a new field. We're moving into a new field, and I think we have to allow ourselves that we don't know everything. We have to invest the resources and the discussion and the forum to understand where are we going, what do we need, and what type of methods do we have to develop for this setting. And many of you have been really, really instrumental also to come to our events. We've had an exchange now for, I think, close to 10 years. And I think it has helped both sides, for us to understand where are the issues in the measurement, but also for others to study and to witness what matters to patients. And I actually think that that is a fantastic opportunity that we have here that helps everyone going forward. And I actually think that this is one of the most important pieces of what you call patient engagement here from the European Medical Agency that we appreciate very much from our side. And I'll stop here for now.
Thank you very much, Bettina. The next member of the panel is online again, Joseph Capalieri from Pfizer. Yeah. I don't see him.
Hi, Joseph
I hear you.
Thank you, yes.
Yeah.
We're curious about the surname Capillary that has roots in southern Italy, Reggio Calabria, the
Not
Ionian.
in Germany. No, no.
Not a German name, and we're looking for diversity, right? And my father was born there, so it's nice. It's really a pleasure for me to be among you. I like to echo in stereo what we've heard by our previous panelists, the international regulatory representatives, HTA representative, patient representative, as well as I'm sure the academic and clinician representative, that we're all, from the industry perspective alone, won't cut it. The industry perspective needs to take into account all perspectives because these perspectives are inextricably linked. Therefore, what we heard, I embrace. I like to complement and supplement the voices about standardization. We already have much to be proud of that's been published in the literature. For example, with the CISA-QoL, the International Standards in Analyzing Patient-Reported Outcomes and Quality of Life data, and also SPIRIT, the SPIRIT initiative, standardized protocol items, recommendations for interventional trials. Work has been published and will continue to be published on how we could agree to standards. And that's important because that allows us to stay focused and targeted on what's relevant. It reduces the uncertainty of what the expectation is from, for example, a pharmaceutical sponsor. So having that standardization, capitalizing what we already have. Now, and here's the next step, providing a combination, a transparent and easy to follow format to put together all that information into one whole. In doing so, we could emphasize case studies, get more specific as to what the details are, the do's and don'ts, so to expand on what we already established and to refine it by matters that may be considered novel and new that we're going to be hearing today and at future meetings. Thank you.
Thank you for that. I think then we move to the last panelist, and that is Christopher Booth of the Queen's Cancer Research Institute in Canada. Christopher?
Hi, good afternoon, everyone. Thanks for including me. I'm a medical oncologist here at Queen's University in Kingston, Canada. I look after patients with GI cancer, and I've been doing that for the last 20 years. And I also run a health services research program that is interested in the big picture issues of cancer care delivery when it comes to access, equity, disparities in care and outcomes. So I'm wearing the hat as an academic and a clinician today. And just to echo the comments, the nice thing about going last is I can let all my colleagues say all the really important things, and just to say I wholeheartedly embrace the comments we've heard thus far. From a clinician's perspective, I think there is important work to be done to make this information accessible for clinicians in routine practice to understand published results of PROs and quality of life so that they can have meaningful discussions with patients. And I think that from the clinical care delivery point of view, that is probably the greatest challenge is, I think in the last decade, our field has come a long way for clinicians understanding overall survival and Kaplan-Meier measurements and hazard ratios. I think the clinical community is also beginning to understand some of the challenges inherent with clinical trial design and some of the advantages and perhaps risks of some of the surrogate endpoints that we use. I think the next real topic that needs to be tackled for clinician engagement is to streamline and make PROs more accessible for practicing clinicians to understand what they're showing, but also to understand what a clinically significant difference is in many of these instruments that can be associated with a statistically significant p-value. But we need help in the clinical community understanding that from a magnitude of benefit perspective. So ultimately, we can have informed conversations with the true stakeholder, the most important stakeholders of this work, which is the patients and their families. So I look forward to the rest of the conversation during the session today. Thank you.
Okay, thank you all for your initial statements on this. It's difficult to wrap it up, but what I hear from all statements is that that basically if maybe if we, EORTC or whatever, can provide robust data on things that really matter to patients and we report them well, we would come quite a way to implementing them for regulatory decisions. And if in the ideal world, if that would be the case, would it be possible for EMA to do that? Or the other regulatory bodies? Who wants to comment on this? Anja, you're laughing, so.
No, I'm laughing because nobody's going to commit at this moment. I think it's not about commitment, it's about recognizing. It's like being an alcoholic, you have a problem, you recognize it, then you can move forward. And I think we've been recognizing the alcoholic problem long ago. And I think, we have a couple of EMA people that know how long this discussion has been going on, what place PRO data should have in the different steps. And now we've come to the end of the story. We had the group therapy. We have agreed they are important. We have also agreed they might be of different importance for different stakeholders. But the important part is that the first one in the row, which is the EMA, has recognized that they can help the others. Whether this leads to including them in a CHMP decision really in a very, very different way, that's not the point. The point is that we have understood the line, we have understood the different stakeholders, and now we want to support each other. That's the end of every group therapy session, I think.
Oh, yes, I very much wholeheartedly embrace that. This is Joe Capillari from Pfizer. I had the HOR, the Health Economics and Outcomes Research Statistics Group at Pfizer, where I'm an associate executive director of biostatistics. But my work is not only through the lens of biostatistics. Is, as we heard, it's taking a multiple stakeholder approach that includes statistics and methodology, but goes beyond it. And that, for those of us who are trained in one area, it's a challenge to understand other people's perspectives and how could one use his or her specialty to accommodate the lens and needs of other perspectives and stakeholders. So there's that flexibility and versatility that comes in what we do to capitalize on our specialty, but don't stop there. Let it expand into an interdisciplinary perspective.
And Jaap, I think I misunderstood your question, but if the question is, are we committed to using patient-reported outcome data for our regulatory reviews? The answer is yes, and it's been a commitment we've had for 5 or 6 years as we've looked to try to improve the utility of the information for our reviews. And I can say unequivocally that the data quality has improved since I began looking at it 5 or 6 years ago. It's not uncommon to see in our commercial clinical trial data sets 80-90% completion rate over long periods of time. Our reviews are consistently looking at the data, mostly to inform tolerability, because as one of the speakers said, and I think eloquently, we really need to have a clear objective, and that's not often so clear in the trial. But I will say the most important objective across all trials, or an incredibly important and consistent objective, is safety and tolerability. So we can always look at the data descriptively to see how it informs the safety and tolerability of the drugs. And we've been doing that in all the reviews where we have patient-reported outcome data at our disposal.
Thank you. Bettina.
I think before moving to the regulatory decision, I would like, and it was mentioned on several slides already, that data is still an issue or complete in the quality of the data. And I think one part of the issue is that someone has to report actually these PROs and someone has to collect the reports. And a part of the problem is that neither patients nor clinicians tend to be very keen on either or. So often the ones, as we just heard, is the clinic, the pharma-sponsored studies where they pay someone to chase the PROs that have complete data while others don't. And it so happens that I just organized a community advisory board on the multiple interests around quality of life and the peer-related PROs from different perspectives. So what matters to patients, what matters to clinicians, what matters to regulators, what matters to HTAs, what matters to payers, because we all talk about it as if we meant the same thing, but it does not mean the same thing. And I think if we really truly want to move, we have to find that overlap in the areas and understand that we want to use the data for different purposes and then try to find a good enough compromise at that setting. And two things that I actually think when it comes to PRO use from a patient perspective and a clinician's perspective is that came out very, very clearly. Patients are not interested in filling your PROs. Patients are interested in their quality of life. And that means that the report has to lead to action that directly improves the quality of the life of the person who is doing the report. So we have a feedback loop that is often not closed. And our clinicians were very clear on it that they would like to have information that informs their decision-making. For example, do I have to alter the treatment? So they were very, very keen on having not just the report, but also the impact. And thankfully, your question, a library had quite a few items that one can add to increase the clinical value of the report itself. So I think we miss an important piece right now because that will give us more granularity into what do patients really value, what do clinicians use to change a treatment. And that, I think, in itself has information value for later decision. So I think we're just too focused and too stuck on the tool. If we miss the upstream part, I think that's a mistake.
Thank you, Bettina. I believe it's important that we would try to go a little bit towards what can we then answer with these PROs. What are then the important places where you feel, okay, now we have collected these PROs, and yes, it is complex because as Pierre started sketching that we have very different scenarios. You, Jaap, explained that in trials, if you look not specific on how you define, let's say, when you collect the data, if you collect it at the wrong point, it has an impact. Carla showed that it's often not landing into the SMPC or in the EPAR discussion. So question now is, what do you feel is, where should we go for? What is it really answering, this PRO? So if you could reflect that, then we can start talking about how are we going to get there. Anja.
I mean, we heard it already several times. You have to start at the start and then you go to the very end. You know, children books are sometimes really straightforward. As a statistician, I have to start with the famous estimate discussion. Even Pierre knows that by now. And that means that I have to sit down with everybody downstream and have to discuss what do we really want to achieve. And a PRO, just as any trial, might not be able to work for everyone and all the questions. And then I have to make hard decisions. Who do I really want to collect these data for? And then when I have made these decisions, then I also have to accept that there are gaps. Maybe it isn't really for the patients in the end or the prescribers, because I can't. In my trial, I might not be able to do that. What else could I do to fill that gap? That's the never-ending story that we HTAs keep telling the industry. You have to fill the evidence gaps that you cannot bring us with your clinical trial data. And PROs are not different to any other clinical outcome. Yeah. So you have to plan, you have to learn to word the scope of your study. We are no longer doing it for measuring the effectiveness and safety of drug X in Y. We have to formulate in which setting, for which patients, in which constellation, what kind of healthcare system, blah, blah. It has to be a real story. And when you do that and you sit down with other stakeholders, like patients, then they will tell you, yeah, you're measuring something that might be good for the regulators, but it's not enough for us. And then you can have this open discussion. How can we make it relevant for you without compromising the regulator or the HTA in between? Then you're moving forward. But that only works if you sit down and you really spell it out loud. Because then sometimes you also will have people that say, yeah, that sounds nice from your perspective, but it makes no sense from my perspective. Yeah, so please try to explain better. And when you try to explain someone else who walks in different shoes, has a different perspective, why you end up with something, then they can tell you why it might be not the smartest thing in the world, how you're approaching it. And this is how you make progress in this field. And we always made it like this, the progress. It isn't specific for pros. It's just that pros have notoriously been the kind of thing that, you know, some of us still remember the reflexes. You can't rely on pros. It's rubbish data. It's never good. And we just heard it, and I totally agree. It has gotten better because we want to use it. And then the last step is showing that it made the difference. The day that CHMP announces we approved this drug because there were doubts, but the pro data were the convincing last piece of the puzzle, you will get every patient and every clinician to sign on to say, well, oh, pro data, we have to make sure that they are of good quality because they can make the difference. And the same holds for HTAs and payers, because that is a proof of the pudding. And are we there? I guess my neighbor says no.
Yeah, I tend to see opposite opinions.
Yeah, obviously, but I mean, at least we have something to strive for, because life is boring if you reach the goal. We all know it's the journey that counts. So let's have the same spirit here. We want to achieve something. We are not yet there, but it is really important to understand how we get there. So let's move forward and be proactive in having these conversations. And
Bettina, you're going on a trip with Anja together?
Well, I just don't like altruism as or moral kind of obligations on people who already have cancer, for example. We will all say, of course, we collect everything. Of course, we never lie. Of course, we do everything right. And of course, we don't. If you're a cancer patient on a trial, you worry about yourself and that's it. So I think the smart way out of it is to deliver a service to the people who have to fill in the PROs and have to collect them and collect the data and do the learning on the backside of it. Because as you want value for you, if you provide value for those who are completing the forms, then you can learn as much as you want on the backside. This is how Facebook makes money. This is how point scheme works in supermarkets. Everyone else does it that way. Why shouldn't it work for us? So I think that was more the point for me, that I would not hope for good intentions or anything. I would rather count on vested interests. And then I think we can learn more, because we've seen it with Bash et al., right? If we saw that they made the ASCO plenary, that you can use something as cause as an EQ5D and even prolong life, just extend life in a very late stage. We now have this already in systems. France is using it for, I think it's called MoveCop or something like this, for lung cancer follow-up, like a personalized follow-up for lung cancer. So it is working, but it has to provide value for everyone in the process. So that was my point in there. I don't like to rely on hope or, moral obligations for people. I think vested interest is much safer.
Thank you. So we're opening up a little bit. So we have, if we just plan it well, have a good study design, we get to answers that even Bettina is looking for. So I think we want to open this up because we see things coming through the web already, talking about the busy clinician, the timing that is needed, the specific scenarios, can digital tools be used? So we're opening up a little bit more for the group here. So please raise your hand. It's difficult to give people on the chat function. We have a couple of other questions there, but I would like to first see if in the room there are immediate responses. Francisco, please.
Yeah, I have a question to the panel, and particularly to Paul Klus and our colleague from Japan. Paul, I think in FDA you had your own journey of, let's say, acceptance and familiarization with this type of endpoints within the regulatory decision. And somehow there is a perception that although the regions are slowly aligning, you might be a little bit ahead. What would you recommend to your fellow regulators from what you learned from your journey of what are the low-hanging fruits on which we can all hopefully soon harmonize, and how we can all progress in a similar way? What are your thoughts here?
Thanks, Francesco. Well, I think we've come a long way with respect to incorporating the data into our reviews. And I think probably the most important thing is moving away from a very black and white perception of the data as needing to show comparative benefit. You know, that's kind of where everyone likes to go, especially in a randomized clinical trial, to say, you know, drug A's effect on quality of life, you know, it improves quality of life versus B, and look more at it as a supplementary piece of information to inform the safety and the tolerability across the board. Where there are no clear pre-specified patient-reported outcome comparative analyses that are statistically tested, and that, unfortunately, and I think most people would agree, is not that common, especially in commercial clinical trials. So, you know, I guess to counter Anya's point, I don't think that we're going to have patient-reported outcomes change a regulatory decision all the time, and I don't think it's necessary to do so. I don't think safety data changes our decision all of the time, but it's always incorporated into our decision in order to assess the risks and the benefits. I do know there's multiple recent examples where the patient-reported outcome data helped us determine that there was a favorable benefit risk based on showing that it was a bit more tolerable, looking at the descriptive symptomatic AE data. So, you know, I think what we've done is moved away a little bit from requiring a statistical comparison across the board to even think about using it, to using it as we use safety data to fit into the context of all the data that's presented to us.
Thank you, Paul. And what about our colleague from Japan, Shun? You have comments on this?
I totally agree with Paul. It is my personal comment. If a similar class of drug is already approved and the study is uncontrolled rather than comparative, PRO may be more useful in examining difference from approved drug. It is my personal comment. I totally agree with Paul. I don't have an additional comment.
Thank you. Any more questions from the audience or remarks? Beate, and Beate first, I think.
Yes, thank you. Thank you. I would like to kind of follow up on the question of how to use that data. I mean, I do agree that of course regulators can support their decision in a way that they feel is appropriate for their decision-making framework. On the other hand, from the HTA side, we do want to actually base our decisions on comparative effects concerning that data. However, I don't feel that this should be any problem because I mean, Anya mentioned the estimate framework. I think it's very well possible in a given study to define estimates for both regulators and HTA bodies. There is no reason not to do that. That just requires a joint scientific advice, and we are moving toward that anyway. So we should embrace this opportunity to, if we set up a study and ask patients to collect this data, to come up with a decision support for all kinds of systems. And then we could also, of course, try to describe that better so that also practicing clinicians could use the data in their discussion with their patients.
I think there was somebody there in the back.
Yeah.
Yes, please.
Yeah, I would like to ask Paul Kloet from FDA, where one of the problems for the regulatory agencies is that the current PRO tools that are being used for quite many years and which are validated, et cetera, where one of the problems is that it doesn't really collect dynamic information, but also the information is not really objective in the sense that, for instance, body weight and length, those are quite objective measurements and there's no disagreement about it. while the pros are often so subjective, and maybe that's the nice thing about it. But I wonder whether in this era we should move on to develop other methods that are dynamic, that are real time, and that really measure the effect on the patient's life. Is he or she able to do more? Is he or she able to go more often to the store to buy? So the activity levels measured in another way than just the answers, whether it's via electronic data or written questionnaire. But is that one of the problems for the regulatory agencies, that the current methodology is just not, that it's insufficient and it's not an objective, measurable information? Quantitative and real time.
Thank you for that question. I would say that I don't think it's an either or. We actually have one study that's going to read out shortly that looks at wearable devices versus patient-reported physical function versus clinician-reported ECOG performance status, right? So like 3 ways of looking at activity or physical function. And we should be presenting that in an ASCO, hopefully abstract shortly. So I think we can use both, and I think both can be valuable. On the subjectivity situation, we already assess subjective information in cancer clinical trials through the lens of a clinician. When we assess safety, I have to go and try to figure out how much fatigue someone is having and then reorient it into my scale, the CTCAE scale, as a clinician. So I think there are going to be subjective information that are going to be important for us to look at for safety and tolerability and for efficacy if it's a pain study. And currently the best assay in my mind for understanding the patient's perception of an unobservable symptom is to ask them. So I do think that currently the assay of patient-reported outcomes to assess an unobservable symptom is best done through a validated patient-reported outcome tool, even if it's a single question as we use for pain, for instance. So I think we can use both. I think there's a lot of excitement about wearable devices and sensors, but I think what we're going to find is the analytical challenges with that data stream are going to be very similar to the analytical challenges that SysACoL is trying to tackle with respect to intercurrent events in the SDMAN framework.
So thank you for that. One of the questions that came up in the WebEx was, and one that we have been discussing also before the meeting, is obviously what Carla Torre also presented, that often PROs are considered not reliable enough, or the information collected with PROs, because the designs were open label. And there it seems that you had a bit of a different view on that, on how that could be handled. So could you please maybe reflect on that, Paul, and then we can go into the differences between the regions also on that perspective. And later I pick you up again, Bettina.
Thanks, and I'll make this very brief. The issue of open label trials is not going to go away. We have open label trials are more common, not less common. And so obviously a single arm trial is an open label trial. So the one benefit of looking at safety and tolerability as your primary objective with patient-reported outcomes is that we don't stop assessing safety data just because a trial is an open label trial or a single arm trial. So, you know, the objective is still relevant there. The challenge comes in when you want to make a comparative assessment. And that should be thought of a little differently. I think that it's going to be, there are some effects that, especially early, that may be challenging. So, you know, the way we handled it in one recent, not so recent now, GVHD assessment, where we did actually look at the patient-reported outcomes in a single arm assessment of improvement in symptoms, so it was very efficacy sort of focused, was that we not only looked at how many patients had an improvement in their symptom, but we also correlated it with clinician symptom assessment, and we looked at how long the duration held out. Because if you have an ineffective toxic drug, even a placebo effect isn't going to hold out for too long, was at least the hypothesis. So there's ways to pressure test that data when you're concerned about a placebo effect.
Okay, thanks, Paul. SysACoL, by the way, is also covering guidelines on single arm trials for statistical analysis, which is maybe important to mention. Let's move on to the second question. Pierre.
I just wanted to express a remark on the blinded trials. Let's imagine that the advantage of the new treatment as compared to the reference is related to a better efficacy or to less side effects. Okay, there I could understand that blinding will bring some objectivity to the assessments. But let's imagine something which is far from rare, where one of the treatments is oral, and for the other one, you have to go to hospital every 2 weeks. What does it mean if you have a placebo injection in the oral treatment? You'd completely destroy the interest of your question by blinding the trial here. So you must make a trade-off here. Imperfect data that mean something are much better than perfect data that do not mean anything. So it is something we have to keep in mind, in my opinion.
It's a very, very valuable contribution, I think, Pierre. So let's move on to the second question on the screen, which is what are the differences among regions and decision makers that require harmonization? Who of the panelists wants to kick off?
Yeah. Hi, I'm Joe Capillari from Pfizer. Some of you may have read an article by Chassany and colleagues published in Future Oncology in 2022, where they go, the authors call for an action to harmonize patient-reported outcomes across key European HTA bodies in oncology. They provided a menu of where the differences are. For example, in with respect to trial design, we mentioned open label studies. In Germany, open label studies are said to be of limited validity, but acceptability in situations when blinding of physicians and patients is not possible. HAS in France believes that such studies are not acceptable. Double-blinded conditions are required for demonstrating improvement in health-related quality of life that leads to clinical added value. And in NICE, no guidance is provided there. With respect to another feature, PRO data collection and reporting on post-progression data collection. In Germany, data collection is preferred until dropout or death. In HAS and NICE, there's no mention of post-progression PRO data. And finally, with PRO data analysis, and these are just examples of several that could be given as to what the distinctions are among the HTA bodies. With the PRO data analysis, missing data sensitivity analysis, in Germany, sensitivity analysis is used to explore robustness of missing at random assumption. There's no analytic method in HAS in France, and in NICE, sensitivity analysis needs to be used to demonstrate the use of alternative utility values on EQ5D data when available from both the trial and the literature. So that's just a warm up. I'm sure we'll have a robust discussion that follows.
That's quite a warm up, Joe. Anyone else who wants to elaborate on this or provide solutions? Anja.
I'm not sure if I can provide solutions, but what
you're up to it.
I'm always wondering slightly if we want to align something, we should be really careful, because if you and Cornel probably can relate to when I say words are very sensitive. So in the physical consortium, we have patient representatives, people that do research on PROs, and they were very sensitive to explicit wordings. And I think the EORTC is very, very familiar with the problem that start coming up when you try to translate a tool into another language. And that's just the language. Then I'm not talking about, if you want alignment, are you trying to remove the cultural, regional differences? Are you ironing them out? Or are you capable of picking them up? Because there's a difference between the technical aspect of the data, the content of the data, and how I acquire the data. And for me, it's very important to understand the differences here, because what I need is alignment on the essence of the data. like blood pressure. You can measure it in many different ways, but in the end, I have to agree on something that we can see as an agreed on international standard on blood pressure that I can derive from different ways of doing it. And for pros, this is absolutely essential. It's so sensitive to say, I did see the word already today. Which one was it? It's the other one, compliance. I saw compliance. Yeah, that's the usual reaction. I was already years ago told, please talk about adherence. I know that the word blinding is totally standard for our statisticians. We have no association of any kind with it. No, please call it masked in some indication. This is so sensitive, and this is just the tip of a giant iceberg. Then how? So what do you want to align here? That's. I was wondering when you asked this question when I read it. What do you want to align? And are you aware of that alignment can actually be detrimental in this context? Yeah. And be careful what you do.
Yeah, I think compliance was my fault in my presentation. Sorry about that. But you suggest to narrow it down to standardization of the provision of the data at some stage.
Yeah, just be careful that what do you want to align? What do you want agreement on? And be careful that you don't lose something that's absolutely essential, in particular in pro data.
Yes, thank you very much. I also would want to support Anja's intervention because, I mean, for example, only a few years ago, we were discussing alignment between regulatory and HTA. I think by now we have understood that we simply have different questions and therefore can't align everything because the question regulators are asking need different information and different studies and whatsoever. And now the same is coming up for alignment of PROs or even alignment between HTA bodies. I mean, decision-making by HTA bodies is so very much influenced by their healthcare systems that aligning everything would result in potentially inappropriate decision-making in individual systems. So I would agree with Anja that we should, first of all, avoid the word align and maybe try to positively try to describe what we want to achieve. That is a standardization maybe that improves quality of data or quality of analysis or reporting. But really alignment seems to be a wrong expression for the whole thing.
So I think also on the question, it was much more, when we prepared this, obviously we were much more correct in how we phrased it, but there's indeed how can we come to harmonization of obviously the different regions. And I think the question is more like, okay, so we can cut it up in pieces. It seems that we have an harmonization of how we go about formulating the PROs and really testing that there are differences between regions, places, countries, people, and the part in that we can harmonize perhaps. And I think the earlier speaker, Joseph Capillari, was more pointing towards what do you want to see? And I think it's coming back perhaps also to we've had the word now, the estimate. Can we phrase different ways of what do we want to show with the PRO? So how to define the research question that may answer the questions of the different groups in need of that. And I was thinking not having that just for the regulatory, the HTA, but perhaps also for that professional and the patient. So maybe I could ask Christopher and then Bettina on what are these kind of clinician and patient views on what do you want to see in the data and what, so that we can also move there to, okay, are there more questions to be answered?
Thanks for that question. I think, in follow-up of my earlier comments and also some comments made by colleagues on the panel, I think that there's a knowledge translation piece of work that's really important here. I think that the PRO community and the experts who have designed and validated and sort of the methodology of these tools have come a long way over the last decade. I think we need to conceptualize not just how this can be used by regulators, but the kind of knowledge translation piece into the clinic. And I think there's a huge amount of confusion amongst clinicians. I know that the PRO community is keenly aware of this and actively working to simplify, streamline, improve education in this realm. But I think this needs to start probably in residency and fellowship training, where there's a lot of emphasis on overall survival and other endpoints. And I think we need to do a better job as a community to teach the clinicians, because admittedly, either the data then does not come out in the primary publication and it comes out later, or if it does, it's extremely difficult for clinicians to understand, given the complexity of both the methods and as well as the number of tools and instruments, as well as the effect size, and then how we can most effectively convey that information to patients who are using it to make clinical decisions. So I think that in addition to the methods and then regulatory considerations, there needs to be this closing this gap in knowledge translating to the stakeholders, both the clinicians and the patients.
Thank you. Bettina, short, and then we go to Paul again.
Yeah, actually, that was my original point. So something that I would like, I don't know harmonization, but I think maybe rather some consensus is like, if you think in oncology, our treatments are moving earlier and earlier. And definitely in melanoma, is like what we've seen in the last decade, and that's my cancer, so my examples always come from there, is like we went from a situation where everyone was dying, and we would have taken anything and accepted any toxicity, to something where half of our people survive, and all of a sudden the discussion looks very different. And we have actually done it in preparation of an MCDA, we have looked at the domains patients consider important, and outcomes and survival is something that patients value. But then we had burden of care, which I have missed a little bit in the discussion here. So on that comment, oral versus infusion is very valid in this context. But they also made a distinction between acute and chronic toxicity. And we often hear talk about PROs collected in a clinical trial. But if we now think about a setting where our therapies move earlier in a disease and we treat, let's say, a stage 1-2 patient who then survives with a low long-term quality of life that on our scales doesn't look so impressive. But I can tell you patient communities are full of people, we have discussed fatigue earlier on, who suffer from things that look minor on paper, but have a huge impact on people's lives, to job loss, things like that, relationships breaking up. So the social consequences of that are huge, also socioeconomic ones. So I would like to see some thoughts on some concepts, how we think about that long-term toxicity, how do we get at the data, and how we can integrate it. Because surely if we had a treatment very early on that had no long-term toxicity, that's better than one that has. So I would like to see some solutions.
Paul, please.
Yeah, I think to Bettina's point, one of the incredible challenges we're facing, and is a challenge to I think Beate's suggestion that we look at patient-reported outcomes serially past progression through to a distal event, is how many sequences of therapies we have now. And so what you think might be in follow-up a long-term toxicity of your first-line therapy may in fact be acute toxicity of your third-line therapy. And so it's become harder to look at long-term toxicity, although I completely agree how important it is. I just want to make the point that, I do think we have an opportunity to meet with HTA and regulators to think about what each different question is and come up with some compromise on particularly the clinical trial design challenges that differential opinions on or differential objectives may create. Because when they are created, and I've seen this, it can result in what could have been a 25 question patient-reported outcome assessment, high quality, more frequently, to a 100 question assessment that might degrade the quality to where no one gets a great answer to their question. So I think there's a lot of room, honestly, still for us to meet and get a little more in the weeds than maybe we had previously on, what would be a common utility that could be used with what we also could imagine being a good patient-reported outcome strategy, maybe the F17 with item selection. I think there's a lot of practical things we could possibly compromise on that would help sponsors, that would help patients, and get us better data.
Thanks, Paul, for your helpful comments, as always. Short comment by Anja, with the emphasis on short.
Yes, Paul just made a very good point, because you just described a situation where the question is coming from a patient. is it relevant to collect data across several lines and then say it isn't relevant for the question of my product, which was first line? No, it isn't relevant for your question for your product or the industry's question on the first line effect of their drug. But it is a question that the patient and the clinician wants to be answered. So they want to know whether third line is worse in a sequence, for example. And if you ask that question, then you have to ask who has to actually collect this data with that specific question in mind and that interest in that question, because it cannot be the regulators, because regulators have to approve the drug and they have to approve the next drug in isolation again. HTAs have a similar problem, but they are not totally free because we are supposed to actually have the entire treatment algorithm in mind. And the patient definitely and the clinician wants to have this data. So the problem is Beate said it nicely, we shouldn't stop at the progression. We should never stop. Patients that enter a trial should be followed for the rest of their life because that would give us essential information. And all of you should actually every 6 months have on your mobile phone the app for the EQ5D, and we should collect it on every single person every 6 months, because then we would really have data we could work with.
Anja, I think you're moving into my area of big interest, because I think it's not possible to collect that or it's probably unlikely to expect that this will happen in the context of a trial. So we need to.
We are not talking about a trial that in the conventional sense. We are talking about something else. Let's talk in the context of the European health data space.
So that's a different story, and I agree with that. I don't think you can answer every single question that everyone has in the single commercial clinical trial that's going to be conducted. Where is the opportunity in actually learning healthcare systems, as I think you were suggesting, Beate? And I'd be interested in Bettina's response to how she would feel with enrolling in a clinical trial and having post-progression patient-reported outcomes.
Okay, Bettina, brief, because then we go again around the room.
So you're impossible. You told me before I'm not allowed to talk fast. So, I mean, you just, so I think I'm just coming back to my original point. If we're able to provide a service to patients, and that Bashar Dal that was at ASCO, that was so impressive. And we've asked patients, and they said, if I could have an EQ5D and I do this once a week, and I know if something goes wrong, they will call me in and they will check me up. And if I have a progression, someone will take care of me, like an extra insurance. They found that motivating. But just for someone else's publication, patients couldn't care less. So I think it is getting that value argument to the one who answers the thing, right? And I stop there.
Good. Well, let's. Okay, there's a question from the audience.
Yes. Yes, I was going to say that before you said it, actually, Bettina, that I mean, if the reporting in itself means something for the patient every time, then it will be useful to the patient. And there are actually today already electronic healthcare system that has, patient reporting in their different domains of symptoms in red, yellow, and green, and the physician at the next visit can really quickly see, oh, this patient has a problem with diarrhea, this patient has a problem with pain, and go directly. It's really useful for both the physician and the patient, and that would, of course, be done throughout the patient's disease through all different stages. And then we have the European health data space that could perhaps collect this type of data in the future. So I'm quite hopeful about that, actually.
Thanks. Now we need to move forward. Sorry, Bettina. We're moving to the last question. We have exactly 3 minutes for that. So that divided by 9 people, that would be 15 seconds for an end statement of. So who wants? What concrete next steps would you propose? Let's start with Maxime.
I think my answer to the precedent question applies here. I think I saw the values of harmonization. I think it's not true that regulators and HTAs can't agree on a couple of things, and I think that the quality of the data is definitely something we can agree on. The ICH-E9 estimate framework is a start, and I think SysQual, I have high hopes for SysQual also will allow us to get common methodologies for handling also the intercurrent events, missing data. There's a lot there that I think we can agree on, and that's a great starting point.
Thanks. Friedrich, may I ask you?
I mean, you're asking for, I totally agree that there are certain things that we could agree on, but you're asking for concrete next steps. So, I mean, an outcome of a workshop like this could be some form of agreement on a journal paper, for example, also in view of more physician engagement, to just agree on maybe certain points within the group that is in the panel that did some talks. So that might be a concrete, realistic next step.
Thanks. Shun, do you want to comment on this? Ideas for the next steps? Okay, let's move on to Harold.
Following Friedrich here.
I'm sorry.
Find an appropriate group that can define what is common ground and what not, and build on that. So taking up what Friedrich said, over.
And Shun is there as well now online again.
Sorry. I would aim to have the result of PRO endpoints included in assessment reports and labels whenever possible. And I would like to, in Japan, discuss more about PRO tools, analyze standard, and the magnitude of effect and PROs that are considered clinically meaningful. Thank you.
Thank you. Christopher, you're still.
Yeah, I think we've touched on some good points. I think that an output would be some kind of educational tool, whether it's a manuscript or some kind of short course, where it would help the practitioners in the field have a better way to appraise PRO data and to understand how to translate into their practice. Similar to many years ago, the user's guides to the medical literature that came out of McMaster University and evidence-based medicine to appraise clinical trials largely with regards to survival endpoints. I think some kind of short course, and these already do exist, but I think maybe drawing attention or raising the profile of some of these resources for clinicians to become more familiar with what is a complex, but at the end of the day, one of the most important things that we can do.
Thanks, Christopher. Joe, you're still on the line?
Yes, I am, and listening attentively and with enthusiasm. We could channel through SysQual the HTA harmonization of the different entities so that we can have more uniform and explicit cohesion and integration, commonality among how to address different characteristics of clinical studies. I endorse the educational aspect with respect to SysQual. That would require sustainability beyond the end of SysQual's life, which formally will be June 2025. We do have a sustainability on education, which could go beyond SysQual to reach other stakeholders and individuals who were not involved. And having publications and short courses would be helpful. Lancet, JAMA come to mind in terms of educating physicians about the merits of patient-reported outcomes, the do's and don'ts, so that we can have a holistic incorporation of the research methodology and clinical avenue.
Thanks very much. Some sort of SysQual part 3, almost. And let's move to the last two panel members, Bettina and Anja. Who's first, Bettina?
So I think I would like to go. I think we're not quite at the end yet. I think we have still intellectual and conceptual building sites. So I think having something like an overview of which areas require more work and where are we not quite there yet. For example, I liked very much your representation of looking at total burden, because at the moment we just have punctual assessment. How do we get a picture of the total burden and link that, for example, with this long-term impact? That would be one. And I'm also interested, so more upstream conceptual and on more downstream. So if we have an outcome, a measure, what happens to it? So what is the value of having this measure? What does a clinician do with it? So I think I want to go both more up and more downstream at the same time, because that is meaningful for patients.
Thanks. And the final word for Anja.
Let's be pragmatic. Make it 2 days next time, and lock the door and don't let us out before we have actually made real differences.
Yeah, good. Thanks. Well, we would like to thank all panel members and all people in the audience as well for contributing to this interesting discussion. Let's now move on to, you want to say something?
Yeah, I want to say you have 13 minutes, so run outside. Grab a coffee, run back in, because we started at 5.
So the last session of the day is tackling novel approaches to the measurement of health-related quality of life and other patient-reported outcomes. And this session will be chaired by my colleague, Madeleine Peer, who already has been introduced herself earlier this afternoon, and by myself, Chantal Quinten. I work here at the EMA as a statistician with a keen interest on PROs, and I also have supported it in the previous years, the EORTC, and more specifically, the CISACOL IMI project. And we're going to, compared to session 1, we're going to continue a little bit further in session 2 to a more like, okay, in session 1, we have a kind of assessed the added value of health-related quality of life for regulatory decision-making. And in this session, we would like to kind of go a little bit more deeper in how to capture health-related quality of life. As was mentioned previously, a PRO has to be useful. So in order to select the PRO, it has to be meaningful both for the clinician, the treatment, and also for the patient. And I think in this session, we focus mainly on the selection of health-related quality of life measures and items for oncology patients. And the first presentation is by Morgan Skronfeld. And Morgan, he's a professor in palliative care and PRO assessment at the University of Copenhagen and also at the Frederiks Hospital. He was a former chair of the Quality of Life group meeting, so he has a broad experience in PROs.
Yes, thank you very much for the introduction. And in addition to what was mentioned, I have no further conflicts of interest than being intensively invested in the past work of the EORTC group, Quality of Life group. So therefore, it's a great pleasure to give a brief overview of the strategy of this group in relation to the development and implementation of PRO and HRQoL measures for cancer clinical research, as we're saying here. So we all agree that we need these fit-for-purpose instruments, but what does it mean and how has this been dealt with and how do we achieve it? We in the EORTC group has had 2 main approaches, one going up until 2016 and a new and quite different approach was built on the first approach from 2016, and I'm just going to give an introduction and explanation of the differences between these 2 approaches. So the traditional EORTC measurement approach was based on these very fundamental issues that we wanted to make robust PRO questionnaires. The core plus module approach was building blocks devised from the very start 40 years ago, and the focus was on rigorous development following a common approach which took into account the differences between the countries and languages and so on, and everything took an international perspective. So that meant that we developed the core quality of life instrument, the QLQ-C30, and a large number of disease-specific modules. Typically, it took 5 to 10 years to develop each of these, and we have a large number of these. So together, you could say this was the EORTC quality of life measurement system to select the core and an appropriate module. A very simple and useful didactic measure. Just pick these 2 questionnaires. And using such clear recommendations gives a lot of advantages, and using the same measures, of course, gives this added value when you increase your knowledge about what you're doing. So the core questionnaire covers 30 items covering symptoms and functioning that are generally relevant for cancer patients. It's one of the most widely, if not the most widely used PROM in oncology, in cancer clinical trials, and also in clinical practice. And there's a large number of validated translations. And this many years of work comes with extensive international validation, knowledge about interpretation, MIDs, reference values, general population samples, cross-cultural validation, and knowledge about the prognostic significance, and also about the utility measure we are going to hear about a little bit later. So that's a lot of advantages, and these advantages apply to the modules as well. So it's a large knowledge base that forms the basis of the strategy. So having praised this strategy, I would say, why was this traditional strategy not sufficient? And the 2 main topics here is the fact that content validity has 2 different applications. One is, can we devise some questionnaires that are generally important, and can we devise some modules that are relevant for specific populations? And we can do this. But on the other hand, every time we design a new trial, we will have smart ideas. We will know the landscape this is taking on, and we are typically testing new drugs and having new comparisons. And that means that standard solutions will never be sufficient for new applications. So standard approaches are good, but they need to be supplemented by something else. The second problem was that all these questionnaires were in fact building on the old-fashioned sum-scored approach, where you have a very primitive way of asking and analyzing your data. And in the meantime, we have got access to the newer technology of computer adaptive testing, and that was not part of the original strategy, but could be taken into account. So therefore, the new quality of life strategy from the EORTC adds flexibility in 2 main ways. First of all, it adds the possibility of transforming the core questionnaire into to CAT, which means that every patient will get the relevant items within each domain for this assessment. And the other part is the lower part of this figure, which is the term item lists, which was in contrast to the previous approach of please don't touch our questionnaires. Then this was an open invitation to actively do touch the questionnaires and mix and add questions and items from these questionnaires on top of what you are already doing. So that could solve the problem of the lack of content validity if done in a nice way and in a comprehensive way. So this new strategy was also one that we communicated in writing with and also in oral communication, but this was an example of the writing where we discussed with the FDA and Paul Klutz about how to phrase this way of doing it. And this was the response we got back from Paul Klutz and colleagues, which I think is a very good summary of the current status of this area. In fact, saying that we favor a thoughtful, that's a very important word, combination of static questionnaires and item banks and libraries to create a balanced, flexible and modular approach to PRO assessment to address targeted trial objectives and to accommodate the needs of multiple stakeholders. So it takes a lot of these things into consideration at the same time. And the EORTC library was launched as a response to this new way of looking at the items with the invitation to actually pick and choose between them. and it contains more than 1,000 items coming from all the questionnaires we had. It's accessible for everybody. It's searchable, and you can look at how each of the items have been developed, where they come from, and what are the translations and characteristics. So the other part of it was the more complicated part of it, of developing computer adaptive testing. And the starting point was that when we're using the core questionnaire all of the time, why not transform this into this modern version? So we developed item banks for all of the 14 dimensions of this. And we put down the rules for this, saying that we wanted to go on keeping the same dimensions, so we expanded on what we had. We included the existing items, which meant that we also kept a backward compatibility. So it's an extension, but it's not a new questionnaire. This was very carefully developed and tested in large studies, and that means that we took the 30 items into 260 items, which are now available for measuring the same domains. And I could talk about this for hours, but in brief, CAT individualizes assessments and improves measurement precisions. It improves power and the range of measurement, and it reduces floor and ceiling effects. So you're getting a lot of advantages, and you can also use it for static measurement, where you simply pick a few of these items and then use them for the measurement. So taking this together, we could say that if we had been doing our very careful initial work of saying these are the 8 domains that are crucial in this evaluation of a new treatment, we could see where they could be covered, and some would be covered in the core questionnaire, some would be covered in the corresponding module. We could develop an item list covering the remaining items, selecting them from the item library. And we could decide that, for example, one of these domains would be eligible for a CAT assessment, because we wanted to provide more detail than just one item about this, and to have a very precise measurement, for example, if this was a primary outcome for the study. So taken together, we could say that this new way of presenting the EORTC strategy uses now 5 key concepts rather than just 2 key concepts like core and module. So we are still talking about the static validated questionnaires, which I would say these are the building blocks for the assessment. We are knowing that we're having the possibility of using CAT whenever that is useful. When we're doing this, we're using the specially calibrated items for CAT. And we're now having the item library, which allows us to produce these so-called item lists, which were also talked about previously, and where, of course, the whole validity of this builds on the rigor lying behind this selection of items. These should, of course, not, we shouldn't skip all the good old work and then simply select a few items that we believe are beneficial in what we want to show. But taken together, this is what we can work with, and which is, you can say, a much more complex area of work, but it allows us to cover, and I think probably one of the main problems in previous research, which was the fact that people were reluctant in order to secure that they added the relevant additional items for the new studies. So thank you for this.
Thank you, Morgan, to kind of reflect a little bit on the 44 years of the quality of life department and quality of life group, where they started and where they are today, and also the dynamic actually over the years. The next speaker is Johannes Giesinger. He will give a little bit more deep dive actually in the quality of life outcomes and disease-specific symptoms. Johannes, he's a professor at the Medical University of Innsbruck, and he has more than 15 years experience in PROs, etc. Up to you.
Okay, thank you for the introduction. So I would like to talk about health-related quality of life outcomes and disease-specific symptoms. To add to Morgan's talks, basically, I have no other conflict of interest than being a member of the EORTC quality of life group. So I'm going to start with like how Morgan's ended, basically with an overview of the EORTC measurement system. So it's like all built around the core questionnaire, the QC30. And as I would say, its extended version, the CAT core with the item banks covering 260 items for the same health aspects as the C30 questionnaire. And then, as we heard before, like there are now more than 49 disease-specific questionnaire modules available, basically to complement the core questionnaire and assess those health issues that are specific to certain diagnostic groups. And this is, of course, all part of the EORTC item library that covers basically all the validated and translated items available from the EORTC measurement system. Just to add what we heard briefly before, there's now a scoring algorithm available. It's called the QLU C10D that allows to calculate utility values from the C30 questionnaire, for example, to calculate quality adjusted life years. So pretty much as the EQ5D. And more recently, we're now in the process of releasing linking algorithms that will allow to convert score points from different PRO measures to the EORTC metrics and vice versa, to make scores, let's say, from the promise measures or the SF36 comparable to allow for data pooling and the like. So it's like the big overview. I'd like to focus a little bit on the modules and more specifically on the use and the development of the modules. So the general recommendations we heard previously was like always to combine like the generic C30 questionnaire with the disease-specific modules. And there's a certain increase, as you can see in the chart, in the use of these modules and the uptake of this general measurement approach. So only between 2022 and 2023, there has been quite a rise in studies in a commercial setting, combining modules and core questionnaire, not relying on the core questionnaire alone. There's a large number of modules available, as I just mentioned, but like the top 10 modules in terms of licensing represent over 70% of the actual use. So there's quite a skewed use in the way the modules are being used. Most frequently, these are the modules for lung cancer, multiple myeloma, and colorectal cancer. And what's also important to emphasize, we heard about static questionnaires like evolving treatment options. So like the side effects of common treatments change over time. And we try to keep up with that also like in terms of the modules. So every now and then, let's say we release updated versions of the modules that try to capture these more novel treatment impacts. And more recently, we, for example, released updated versions of the breast cancer module, of the lung cancer module, and the head and neck module. As we heard like earlier, it's a time-consuming process, so it's not that flexible. It's not that up-to-date, maybe. That's why we have like this additional option of complementing the modules with additional items from the item libraries to be like more like to meet current needs more quickly, let's say. The development process itself is like a multi-phase process, and it's like basically kind of the foundation for the quality in the end that we might get from our measurements. It starts off like the creation of the content based off input from patients and healthcare professionals. It's followed by like converting the content that is relevant to a certain target population into items, to conduct like a preliminary testing of the items in terms of patient acceptability and of the completeness. So we like revisiting the work of phase 1, the content generation, in phase 3 to see if any important health issues that patients consider important are still missing in our questionnaire. This is followed by phase 3B, preliminary assessment, let's say, of the measurement characteristics of validity, scale structure, reliability, usually in 100 to 300 patients, depending, for example, on the questionnaire length that we are evaluating. And finally, in phase 4, when developing modules, we do like basically the full validation of the questionnaire. Usually in more than 300 patients, we're assessing reliability, scale structure in a confirmatory manner. And among other things, very importantly, we assess sensitivity to change of the questionnaires, which is like a crucial characteristic of our questionnaires in a clinical trial setting. So it's like the multi-phase process that underlies the available disease-specific modules. It takes, as we heard earlier, quite some years to do this. It involves at least 500 patients. Very often it's way more than 1,000 across all development phases. And what is also like very important, like being a European organization, we have a strong focus on cross-cultural validity, on translatability. So from the very start, we include patients from different countries and more specifically different language groups. So as we heard, like the source language we're developing our questionnaires in is English, but then again, many English terms are not easily to translate to the different languages we have in Europe. So we try to consider this from the very beginning. We do translatability assessments to avoid certain specific words that might be problematic later on in the process, let's say. So this is like followed throughout the process, let's say, this assessment of cross-cultural applicability and validity. Maybe a little bit more detail on phase 1, because it's so crucial, is basically the phase where we define the content that we include in our modules. This is very much, as I said, like about patient input, like patient interviews, input from health professionals with different backgrounds, physicians, obviously, but also psychologists, nurses, and social workers. So we have try to have like a reflection of different professional groups involved in the treatments of the patients and their views on it. It not only includes like an assessment of what patients consider like what they experience in this diagnostic group, let's say, but it's also like an exercise of prioritizing based on patient input. Basically, let the patient prioritize what should be in the questionnaire, because it can be like very long, and there's always a certain trade-off between the length of the questionnaire and the relevance and the content coverage. So we try to have patients prioritize what should be in the final questionnaire at this early stage. So that's like all about the focus on content validity, basically making sure that the questionnaire assesses what matters to patients. Patient representation is like also like trying to have like a good coverage of the various subgroups of our target population. So for example, if you develop a module, let's say for breast cancer, we try to have a representation of different subgroups, for example, defined by treatment type, disease stage, etc. And this sampling matrix that we kind of define at the very beginning of the process is followed through the whole 4 phases to guarantee like a representation of different types of patients when determining the content and assessing the validity. So on the next slide, the C30 has been like developed way early. As we heard, it was published in '93. Like this procedure that I just mentioned was not in place back then for the core questionnaire, but only later for the modules. So more recently, we did like a study to see if the content that is covered by the C30 is still relevant, if it actually the core questionnaire provides content validity and is measuring what matters. This was like a qualitative study in various different types of cancer patients, and was about like concept elicitation, so to find out what really matters to patients on a broader level across diagnostic groups, across treatment types. And it was kind of reassuring that the 13 most frequently elicited concepts are actually covered in the core questionnaire. That was kind of reassuring. But of course, there are also things missing. So for example, health issues like raised by patients that were not covered by the core questionnaire, but are partly in the modules, are, for example, climbing stairs, weight loss, which might be like also objective measurement, as we heard earlier, skin problems, numbness, etc. So there's quite some evidence for content validity, but of course there are things measuring, which also highlights the need to complement the core questionnaire and not rely on it only. At the very end of my talk, I want to quickly touch on the issue of interpretation of PRO scores. So if you have like administered collected data with the core questionnaire, with the modules, a common challenge that is frequently mentioned by many stakeholders involved in this is like the challenge of interpreting the data that you collect with such questionnaires. So it's very common to have like score points from these questionnaires, let's say on a 0 to 100 metric or other metrics used for CAP measures. And these score points can be very abstract. It's sometimes difficult to make sense from this. And so you need like various types of, let's say, interpretation aids that are differently useful, I would say, for different stakeholders, different settings also. And EORTC tries to provide like a broad coverage of different approaches to data interpretation. So I want to give you some examples. For example, for many of the measures, there are like thresholds available that help you to interpret the clinical meaningfulness of changes over time, of difference between treatment arms. And more recently, there's like more awareness, I would say, for the distinction between thresholds for patient level data and group level data. That's also like one of the issues we talk about a lot in EORTC, EMA. And so you might need different thresholds to define, like if a change of a single patient is like clinically meaningful. That's a different question compared to if like the change of a whole group is different, obviously. And there are different thresholds needed to assess like differences between treatment arms, etc. So that's like a very important point. There's been quite some misuse, I would say, of different types of thresholds for wrong settings. Then maybe more like in daily clinical practice, we need like different types of thresholds that focus like on the individual patient. And if a patient completes an assessment, it's important for the clinician to decide if this is like a concerning level. So as we heard earlier, if like do like a patient monitoring, let's say a remote monitoring of the patient being at home, you need some guidance if a patient reports a certain score in the end to decide if this is something concerning or not. So we're developing cutoffs for symptom screening in some way, I would say. Quite a different approach, not relying on thresholds, but more like on comparative data, to call it like that, is like we have collected reference data from different European countries and the US for general population, which is, for example, helpful when evaluating health-related quality of life in a survivorship sample. It depends on the context what is most useful. There's quite some data available from the EORTC itself on various types of cancer patient groups defined by diagnosis or treatment type. Right now, this is only like a PDF document, a long one, let's say. In the moment, we're like kind of extending this with an online database with new data to make it more accessible and more usable, to have like comparator data from different types of patients to make the data from your specific study population more useful. And in the moment, we're also like working on a manual that will like provide guidance on all of these interpretation approaches. In summary, at the end of my talk, I just would like to highlight there's like a wide range of modules available that are like in a certain way also providing like a core outcome set that makes it easier like to compare what we had earlier, like indirect comparisons across trials, because it's like a fixed set of outcomes you assess. It's less selective, less variable across different trials and settings. There's like quite some emphasis on guaranteeing content validity. So we really try to make sure from the very start of the development of the questionnaires that we really assess what matters to patients. And let's say at the end of the process, we try to support with different methods, let's say, the interpretation of the scores that you get from these questionnaires. Thanks.
Thank you, Johannes, for that fantastic presentation. And so now we're going to move on to a joint presentation between Claire Pichinin and Alexandra Gilbert. So Claire, so if you have used the item library, you will probably know Claire because she is the behind the scenes and the ones that provide support for all users on the item library. And she has been with us at the EORTC for more than 5 years. And with Alex Gilbert, we're competing with how long Alex and Johannes has been at the EORTC as an EORTC member. So Alex is a radiation oncologist and has been an EORTC member since 2011. So that means you're 15 years with the EORTC and working on the item libraries as well. So we're very much looking forward to hearing your presentations.
Thanks so much for the lovely introduction, Madeline, and thank you to the organizers for the opportunity to be here. It's really a pleasure to get to hear from the other presenters, panelists, and participants. And of course, I also have to acknowledge the bigger item library team at the EORTC who helps manage a lot of our day-to-day requests. And so today, Alex and I are going to talk about more flexible approaches to PRO measurement through use of item libraries, some of the work that's been done to support the use of item libraries, as well as what their use might look like in practice when implemented in different settings. And I can confirm that we have no conflicts of interest to declare. So when we talk about item libraries, it's important to highlight some relevant terminology. And I know that a lot of these terms have already been brought up today, but just to sort of expand upon what was previously presented, when we talk about item libraries, we're referring to collections of single items or multi-item scales that measure various PRO and quality of life domains. In contrast to static questionnaires, which can't be modified, item libraries allow for customization through selection of specific targeted items. We can then further differentiate between item libraries that are derived from existing validated questionnaires, like the EORTC item library, the Facet Searchable library, or the MD Anderson symptom library, from those that were designed and developed with the specific aim to create a flexible library of items like the PRO CTCAE. And of course, it's important to distinguish item banks from item libraries, as item banks are specifically designed to facilitate the use, of course, of computerized adaptive forms of testing. The term item list is used to denote a customized questionnaire created using a flexible item library approach. In many cases, depending on the underlying PRO measurement system, item lists can benefit from existing questionnaire frameworks, including previous validation work and available translations. A lot of you will be familiar with the EORTC item library. It's an interactive online platform that serves as a sort of database of validated EORTC questionnaires and their items, supporting the development and validation of new questionnaires and questionnaire updates. It's also an important reference tool, of course, allowing users to search through items using different classification frameworks, browse through questionnaires and translations, and importantly, it allows for the creation of customized item lists for use in different settings. As highlighted earlier, this particular feature, which allows for more flexibility in PRO assessment, is an important hallmark of the EORTC's current approach to PRO measurements. When it comes to EORTC item list creation and implementation, we see different patterns of use between our primary user groups. So the majority of item lists that are created by industry users are used within clinical trials, with a smaller proportion used for observational studies. In contrast, for academic users, most item lists are used within observational studies, followed by trials, and then for clinical monitoring purposes. I think these trends emphasize some different stakeholder needs, as well as the relevance of flexible PRO and measurement approaches, even outside of clinical trials. And when designing and implementing item lists, ultimately it's important to keep in mind the content and the context of use, and recommendations should really be sort of tailored depending on how an item list will be used. Flexible approaches to PRO measurement through use of item lists are relevant within many different settings. As highlighted previously, this sort of approach can help to ensure that the effects of novel treatments, which may not have been available or relevant at a time that a given questionnaire was developed, are captured. For rare disease groups for whom questionnaires may not be available or for whom questionnaire development might be a challenge, item libraries can provide another way of measuring PROs. And within early phase trials, when less is known about possible symptomatic adverse events, for example, item lists can allow for inclusion of different issues, helping to define a PRO strategy that can be updated later on. And I think even beyond these particular examples, item libraries can help to address the important need to be pragmatic and minimize patient burden, which is especially relevant for certain populations. And as Vishal highlighted earlier, I think item libraries also provide sort of a pragmatic and practical way of potentially balancing needs of different stakeholders, including HTA and regulatory authorities. There's a lot of heterogeneity in terms of the content of item lists created using the EORTC item library. As we've seen, they're actively used in many different settings, and ultimately this helps sort of inform how they're created. But many of the item lists that are more frequently used and requested can be divided into sort of a few distinct categories. Those that measure core outcomes, both functioning and symptoms, for example. These item lists are often comprised of subsets of items from the QLQ-C30. And in terms of the specific measurement approach, in some cases, the full C30 may be administered at some time points, and distinct subscales in the form of an item list at alternative time points. Treatment-related side effects are often captured in item lists, which are administered alongside a core questionnaire like the QLQ-C30 and a module where relevant. And increasingly, global impression of side effect burden is captured in a single item list, which asks patients to what extent they've been troubled with side effects from their treatment. And again, this item list is often administered alongside a core questionnaire and a module. And I think that these examples are also very much reflective of the scope of regulatory guidance currently for use of PROs in cancer clinical trials. Since the launch of the EORTC item library, many of the research projects we've undertaken have been aimed at highlighting sort of the coverage of items in terms of PRO and quality of life domains, and of course, facilitating the selection of items and the design of item lists. Within this scope, we carried out a descriptive mapping exercise in which we linked the then 950 EORTC items to the CTCAE framework to evaluate coverage of adverse events. We were able to link 208 different CTCAEs to EORTC items, and more than 65% of EORTC items were assigned at least one CTCAE classification, illustrating pretty considerable coverage of adverse events. These findings have since been integrated into the item library and provide an alternative means of searching for and identifying items using the CTCAE clinical framework as a reference. I will now hand it over to Alex. Thank you.
Please do clap. Fab. So thank you so much for, again, everyone for inviting us. So I'm actually, I think, moved on a previous slide, so I need to go back. How do I do that? Yeah. So I am just going to follow on from what everyone else has said to talk about how we develop a PRO measurement approach. And I'm a jobbing clinician working in clinical trials. So this is also reflective of some of my own experience about how I've gone about it. So one of the issues that is really, really important to cover is those core outcomes of issues which turn up again and again for patients. And probably the safest way of doing that to ensure that you're really covering what matters to patients is through the use of a core questionnaire and then a disease-specific module, because then for sure that you know that there's been a huge amount of work that's gone into the development of those questionnaires to ensure that issues that are regularly relevant to patients are then covered within your clinical trial setting. Then we move on to the issues around tolerability and how, of course, we need to think about how to identify those issues from an item list. So there are obviously, when you're coming up with a novel drug, you need to always think about how that novel drug is going to impact on patients in terms of the tolerability. There's usually a lot of information out there already on it, even though it's maybe not gone through extensive clinical trials. There may be something from another setting that you can access from the investigational brochure, literature, speaking to clinicians, and of course, speaking to patients, representatives as well about those sorts of issues that might come up. There are also, which has come up before, other issues that you may want to consider about how you're measuring overall impact or burden of your particular novel agent, for example, in your new population. And the symptom burden item, I think, really helps define that issue. You may want to, if you have a special patient cohort, like an elderly frailer cohort, again, you may want to consider the use of a specific module, maybe not at all time points, but because you're worried perhaps about patient burden, but you still want to capture the impact on a different population and may want to include it. And I work a lot in an organ preservation setting, so reducing side effects for patients, trying to get kinder, less toxicity for patients in the longer term. And questions around decision regret can be really helpful here. So there's different scales out there that you may want to think about including within your trial to get a view around all different issues. And actually, there are now guidelines coming out around this, so that's obviously helpful for everyone. So Claire led a group to try and develop some guidelines around how we use item libraries in particular, this more flexible use of instruments. And I'm actually now leading a project that's along with Claire on developing kind of consensus guidelines that are going to be finding out from different stakeholders what are the sort of decisions that we need to think about when we're using item libraries within our clinical trials and also routine care settings. So this is a study that I want to just use as an illustration to help see where patient-reported outcomes can be of real value. So this is the Foxtrot trial platform. That's a UK academic and industry mixed study that's funded by the Yorkshire Cancer Research, that is looking at the role of the use of neoadjuvant chemotherapy in locally advanced colon cancer. And I think this is a really nice trial to demonstrate the value of patient-reported outcomes. So this is using targeted agents and also chemotherapy. It's looking at frail populations. And so it's got the whole reach. It's because it's neoadjuvant. You also have to think about the fact that patients are having drugs. They're also going to have surgery, and how that complicates how you're going to measure your treatment over time. And it's also in a curative setting. So you're actually really worried about long-term outcomes as well, because actually, and no measurement of late toxicity is key. So as a clinical oncologist, I'm very used to worry about late toxicity. So I felt like I was up for this task. So here I'm going to focus on Foxtrot 2 just to illustrate the example. This is a frail population. Patients don't often end up having adjuvant chemotherapy. And this is a trial looking at whether giving dose-modified adjuvant chemotherapy is actually valuable to patients and improves their outcomes. So we really wanted to make sure that when we were measuring our patient-reported outcomes, that we captured global aspects around patients' role, physical function, obviously in this frailer patient group. Of course, we were worried about symptoms, but we were also worried about disease-specific symptoms, and also the fact that you are having a surgical treatment process. So, for example, the vast majority of patients will probably have a temporary stoma at some point along their treatment pathway. So you need a tool that's able to flexibly move between the issues that you have with bowel function when you just have a tumor in situ, and when you don't have a tumor in situ, but you have a stoma. And the use of the colorectal cancer module allows that fluidity between those different timeframes within a patient. And also then, of course, if they end up having their stoma reverse, it again allows that longitudinally over time. The colorectal cancer module is now actually quite old. It was originally validated. The most recent update of the validation was actually in 2009. And so actually oxaliplatin wasn't used very commonly, but it is now. And so we needed to include the peripheral neuropathy toxicity, which is hence why there's an item list in that. And again, I mentioned this is a frailer population, so we're including the elderly cancer module to ensure that we capture that the issues that elderly patients may be suffering, but not as many time points as the toxicity assessments. And in the other different trials, which are looking at targeted agents and immunotherapy, again, we've developed the item list that are specific for those different groups, so that those individual toxicities are present as well. But you can see there's a core of different issues that are measured across the whole platform. This is just to highlight particularly the value of assessment of patient-reported outcomes in an early phase trial setting. And I think this is a really excellent piece of work that was carried out in Toronto. They used the PRO-CTCAE to ask patients who are on multiple different early phase systemic treatments to complete questionnaires about their treatment at various time points. And you can see here how important that systematic process of patient reporting is so valuable. As clinicians, we have a different process that we need to go through when we have a conversation with patients even patients who are in a clinical trial setting. We miss things. We don't have a systematic approach to asking 80 different questions in our consultation. And this provides the additional safety information that, particularly when you're trying to estimate the dose that's required, is really essential, particularly if you identify these issues, if there's a global burden of multiple different low-grade toxicities that we often see with our systemic agents now, the targeted agents now. That's really essential to pick that up at that early stage before you go on to a really expensive phase 3 trial. And this just highlights again how allowing for free text, or potential missing of issues that patients may have that you haven't spotted before, because it's a novel setting, how important it is that you allow patients to have their voice as well, to be able to identify additional issues that you may have missed, despite how thorough you thought you were when you were choosing all your items. And we have that flexibility within the EORTC now, and the same process is available for electronic use of the PRO CTCAE as well. So in summary, I hope we've all demonstrated how important the use of a flexible item library is useful in establishing tolerability, how key that the core outcomes are still covered, and how they can actually be used as a measure of efficacy as well, with improvements in disease-related symptoms. In particular, when you have multiple modalities of treatment, so surgery does not use the CTCAE to be able to measure their complications. And so we actually, as using patient-reported outcome data, when you have these multiple modalities of treatment, you have that one measure that allows that consistency of experience over time. And of course, as we've mentioned multiple times, we're measuring what matters to patients, and that's obviously why we're all here today. So thank you so much.
All right. Thank you so much, Claire and Alex, for that presentation on the item library. So now we're going to, I'm very happy to also to introduce Jim Shaw, who is now online. And he will be providing the industry perspective on the use of static and flexible PRO measures in global cancer trials. And the reason we wanted an industry perspective on the use of these PRO measures is because at the end of the day, they have to use it for global trials. So it would be very useful to know what are the challenges that they are facing, or what are the benefits of having these measures used in their trials. So Jim, the floor is yours.
Thanks, Madeline. And first, I want to apologize for not being able to be there today, and I'm very thankful for having the opportunity to present. Over the next 15 minutes, I'm going to try to provide some sponsor perspective regarding the potential of what we see as the benefits of static and flexible measures, PRO measures. Next slide, please. These are my disclosures. Next slide. So I'll provide some very cursory background information. I think some of the topics I have in my slides have been covered earlier. I'll try to highlight what I see as some important points that may not have been covered, but then move into a discussion of the experience at Bristol Myers in terms of using flexible measures and what we see as their relative merits and costs vis-a-vis static measures. Next slide. Next slide. So we're all familiar with static measures of patient-reported outcomes or quality of life. I think what's most important to consider for this talk is evidence supporting them. In particular, static measures, by virtue of their history, oftentimes are supported by a wealth of scientific data on their validity in a variety of clinical contexts, oftentimes much more so than flexible measures, and that becomes an important point for distinction. Obviously, some key measures noted here, the QLQC30, and my personal favorite, the EQ5D. Next slide. I think earlier presenters covered item pools and libraries. The primary distinction here between item libraries and item banks is the latter effectively serving as a vehicle along with a vehicle to inform computer adaptive testing, which is the real novelty, the possibility of using what would amount to briefer, more targeted item batteries to measure concepts and ultimately result in efficiencies in instrumentation in clinical trials. Next slide. So Again, I think that most of this was covered by previous presenters, so I think we can skip to the next slide. I'll reiterate because I think this is important. This is the I guess the key point when at least the key area of focus when differentiating measures based on flexibility. Computer adaptive testing has a lengthy history in educational settings. More recently has been brought into the healthcare arena and the clinical trial space. Obviously, computer-based assessment has a number of advantages, whether used with CAD or not, including immediate data entry, improved data integrity, ease of scoring, and reduced time to availability of results. With CAT in particular, respondents or the items that respondents are presented are determined by a function of respondent ability and item difficulty, so that respondents are only need to complete a small number of informative items in order to generate a summary score. Next slide. So what I think is important to take away, or a key takeaway of what I'm going to present today, is that flexibility is a continuum. Measures can be defined as static, such as historical forms developed by the EORTC, customizable, such as those based on item libraries like the PRO CTCAE, and truly dynamic measures, particularly those based on CAT, such as the Promise CAT or the EORTC initiative that was briefly mentioned earlier. Next slide. So with that, I want to segue into some discussion of experiences at Bristol-Myers using flexible measures, and much of this has involved use of the PRO CTCAE. So what I'll try to do is extrapolate to flexible measures in general where possible. The PRO CTCAE was developed to allow for evaluation of symptomatic toxicities by self-report in adults. Since its inception, versions amenable to use in adolescents and children in cancer clinical trials have been developed. The adult library includes 124 items covering 78 symptomatic toxicities. Items included in the library measure the frequency, severity, and/or bothersomeness or interference of each of these symptoms. What makes the PRO CTCA item library useful, and this can be generalized to other item libraries, is the ability to tailor content to the specific needs of the trial. So, in particular, with the PRO CTCAE, the ability to use a relatively small number of items to provide an unbiased assessment of the tolerability profiles of all comparators under study within a trial. Additionally, I want to point out that the PRO CTCAE utilizes extensive branching logic, not necessarily, not terribly different from the logic applied with CAT, and insofar that does present a potential issue or barrier to using the measure with ECO in trials, and I'll touch on that in a bit. Next slide, please. Bristol-Myers was an early adopter of the PRO CTCAE, so we started using the tool in trials as early as 2016. To date, we've had 14 studies in hematology or oncology indications using the PRO CTCA, including 6 phase 3 trials. We have published on methodological considerations when using the measure, and perhaps more importantly, collaborated with NCI on the development of de novo items for use in some trials where existing items frankly were not sufficient to address the target profiles of some comparators. That highlights the key feature or value of flexible measures. That is, again, the ability to adapt to and address the particular needs of any given trial. Next slide, please. So in terms of our learnings from working with the PRO-CTCAE, I want to try to focus on what I see as general to flexible measures as a whole. When one is developing a clinical outcome assessment strategy for a trial, one needs to align with multiple functional stakeholders and capabilities, and this can present challenges to using flexible measures. Added complexity or movement away from the norm requires a lot of hand-holding and different arguments to address concerns of different stakeholders. Something that adds uncertainty or calls into question prior experience may make it difficult to justify using a flexible measure in a pivotal study. So in cases where we might consider using a flexible measure, particularly one that we might not have experienced using previously in a planned trial, conversations with colleagues in global development or regulatory affairs might focus on regulatory applications of the data. In particular, whether there are any precedents for competitor use of said measure, and whether there are any regulatory precedents, any precedents for data based on that flexible tool entering US or European label. Beyond this, even though this meeting is focused on regulatory applications, I think it's important to consider that any data generated in a trial will be considered for their market access uses. So conversations with colleagues in HEOR or market access would certainly focus on those HTA applications. How the data generated using a flexible tool would ultimately fit within the larger value story, what would be the local HTA perspectives on that particular measure, and perhaps most importantly, whether based on their own guidances, it might be considered a validated tool. Additionally, conversations with colleagues in biostatistics would center on educating them regarding the scoring, analysis, interpretation, and presentation of results based on a flexible tool, placement of endpoints based on that measure within the statistical testing hierarchy. Also, derivation or specification of estimands for those endpoints, and given that flexible measures oftentimes generate more information than static measures, what data critically would need to appear in the CSR, and how those data should be represented either in tabular or visual form. Next slide, please. Also keenly important to take into account operational considerations. Before seeking to implement a what I'll call a novel flexible tool in a planned trial, would definitively want to have conversations with colleagues in ECO enabling capabilities, those responsible for licensing and translation, would also want to consider ECO supplier capability and potential device limitations to using or fielding this flexible tool. In particular, not all ECO suppliers have familiarity with or the facility to accommodate flexible measures. Even enacting conditional branching can be a problem for some suppliers. One must also consider the fact that not every sponsor has the facility or the freedom to work with any and all ECO suppliers. Oftentimes, procurement policies may restrict which suppliers can be used in a given trial. Additionally, all large sponsors are now seeking to improve the efficiency and reduce the costs of their development programs. Companies may well have internal capabilities whose role it is to reduce trial complexity, and insofar as they do, seeking to implement a flexible PRO measure in a trial may run afoul of these groups. Next slide, please. So in my remaining slides, I want to just touch on what I see from a sponsor perspective as the comparative benefits of static and flexible measures. Next slide. For static measures, perhaps the foremost benefit is familiarity. Stakeholders know what to expect and can readily understand the content of the measure and its scoring. The results of inter-trial comparisons based on static measures often may be easier to communicate than those based on flexible measures. the ECOA build in general will be easier with static measures than flexible measures. In particular, computer adaptive testing is a heavy lift for ECOA suppliers, and many cannot accommodate it. There may be unforeseen challenges with respect to scoring, data transfer, or UAT. Even when it's feasible to use a flexible measure with a given ECOA supplier, design processes are apt to take longer than they would for a static measure. Again, highlighting the importance of HTA. Oftentimes, HTA bodies do not possess the scientific personnel with the scientific expertise that one finds within regulatory bodies. And so communications about flexible tools may be made more difficult with them than with static measures. It may not be easy to tell a representative from an HTA that one trial participant can complete 5 items while another can complete 12 items, and they both have the same score interpretation. This is compounded by the demands of some HTAs for quote-unquote validated measures. For health technology assessment bodies, it can be difficult to apply the term. It can be difficult to define what validation means when flexible measures differ in item content, either across trials or within a trial across participants. Certainly, the number of PRO labeling claims overwhelmingly reflect static versus flexible measures. And while this is a function of the time during which flexible tools and CATs have been available for use, precedents weigh heavily on internal decision-making, particularly when timelines are tight. Additionally, regulatory guidance in this area is certainly making up ground, but is still catching up. Additionally, I think it's important to consider content validity, which I was happy to see mentioned several times today. Content validity can be a problem for both static measures as well as flexible ones. However, given the pragmatic sources of item content for many item libraries and item banks, I think it's a particular concern for flexible tools. So if I were to ask my 86-year-old mother who was recently diagnosed with pancreatic cancer, what she would think about some of the items in the Promise item bank, in particular, I think she'd find it bizarre if I asked her if she could pass a 20-pound turkey around the dinner table. Next slide. I don't want to give short shrift to the benefits of flexible measures, but from a sponsor perspective, I think their benefits are much more targeted and in some regards less practical. Certainly the ability to tailor content is the main advantage of flexible tools and potentially allows us to optimize item content for differentiating trial comparators based on efficacy or tolerability. This also allows us to use more targeted questionnaire batteries with reduced content overlap and potentially reduced administration time. This may have benefits for study participants and site personnel, and importantly, may make it easier to recruit sites and retain participants in the trial. For item banks that use IRT-based scoring, there may be other advantages. Referencing scores to a T distribution has some potential cachet in terms of statistical interpretation. And certainly because CAT allows us to focus estimation precision at any point along the measurement continuum, an argument can be made that it allows one to better tailor precision to accommodate the needs of a study and perhaps capture or reflect the extremes of respondability. Next slide. So in conclusion, want to reiterate flexibility should be viewed as a continuum. With flexible assessment, the main advantage is the ability to tailor item content to individual needs of a study, but there are numerous pragmatic barriers to using flexible tools, and in particular CAT in clinical trials, and many of these are not easily addressed by the developer. The application of flexible measures require the support of appropriate sponsor roles, processes, and platforms. Additionally, supplier technology, which may interact in confounding ways with sponsor processes. Last, certainly regulatory precedents for labeling based on flexible tools lags that of static tools. And I think it's important to address HTA concerns or interests regarding flexible measures if they are to be adopted more widely. So with that, I'll be happy to end and hand back over to the organizers.
All right. So thank you so much, Jim. That was a very thoughtful presentation. And I think we're very grateful that that BMS was willing to share some of their experiences in the use of these tools, both for static and the flexible measures. So with that said, we're going to move on to the panel discussion. And my co-chair, Pierre, unfortunately has to leave early because he has a train to catch. So now Chantal Quintin will also be co-chairing this panel session with me.
Thank you, Madeleine, and have a nice journey, Pierre, to the train. We did prepare some questions, and we still have 40 minutes, so let's benefit from the last 40 minutes of today and then the conclusion. But we would kind of like the people listed here to kind of introduce themselves and to share with us first reflections before we kind of pinpoint down the questions that we thought would be of interest. So I think Hans Schreuder, your answer. Yes, Hans.
Thank you. My name is Hans Schreuder, and I'm a patient representative. I'm a myeloma patient, a cancer patient as well. And my root organization is MPE, Myeloma Patients Europe. But also I'm quite involved being a vice chair at the moment with the weekend work group where also Bettina is part of. Well, looking at, I must say a lot of my lawn is already mowed by the former speakers and the former panel. So I thought, what can we add to this? I think one of the aspects that we need to be aware of is not only, of course, the most important thing is looking at the items that are important, standardize that, and also the time you pick up. That's the EORTC project, I think. We, as MPE, have been involved with the EORTC project. Actually, we were the lead of one of the packages. So we have had a lot of experience also to include non-statisticians in this kind of statistic decision making and making considerations. And that was quite difficult, I must say. But I'm really impressed by the EORTC project leads of EORTC that made that effort to include the patient advocates to understand every item we went through. And that's also a bit the thing with the item bank, I think, when you design a patient-reported outcome questionnaire. In the end, it's the patient that needs to fill it in. And so you need also make that step to understand how is it understandable for the patient? Are the questions that way formulated that a patient has no doubt about what the answer should be? The other part is that it's not an item with 100 questions because you want to let a patient fill it in not once, but also over 3 months and then 6 months, maybe once a year, and maybe for many years. So there needs to be a consideration about how can a patient adhere to filling in and giving us those data, because otherwise we won't get those data. That was one consideration I want to pick up here. Thank you.
Thank you, Hans. And then the next speaker, Mogens, we have been introduced, but maybe you want to share your reflections or?
Yes, thank you. Yeah, I had a bit of a chance before, but I think one of the things that I have been thinking about is that we're having these pendulum. When I started, everybody was asking a junior colleague to write a questionnaire and then use it from the next day. So that was the level we started on. And then we spent a lot of years of standardizing and persuading everybody you have to use standardized tools. And then we realized that everything is not standardized and we need to add flexibility. And then we're seeing the pendulum going back to say that we have to save patients for all these questions, and we need to be targeted, and we need to be efficient, and so on. Understanding this movement towards different equally valid concerns, you can say, of addressing all these things. I think that's the overall challenge we are looking at from different perspectives here, and I think it's a collective learning exercise we are in, and we should just acknowledge that in order to learn, we need to be confused at sometimes, and then we can build on. Yeah, thank you.
One of our next panelists, Ashley Wilder, she might be online. Ashley?
Hello, can you hear me?
Yes, we can.
Terrific. Thank you so much. I just want to thank the organizers for including me. What a terrific set of talks today. Just to situate myself, I'm at the U.S. National Cancer Institute. So we're a research organization, and I'm the chief of the Outcomes Research Branch. And the mission of my branch is to support research that monitors, evaluates, and improves patient experiences, health outcomes, and quality of care across the cancer continuum. And one of our main objectives is to develop and evaluate PRO measures. So just to, for full disclosure, not in any conflict of interest way, but just to let you know, an example of our work is being central to the development of both PROMIS and PRO-CTCAE in order to fill needs that were identified in the US related to the capture and interpretation of patients' perspectives about their own health. PROMIS, as most of you know, was designed to address needs for a systematic approach to the capture of quality of life along physical, social, and mental health domains that allowed for comparisons both within and also across diseases, as it is disease agnostic, and over the life course, and in a variety of research designs, as well as to inform clinical practice. And the development of PRO-CTCAE was really designed in the US as a complement to the standard CTCAE, or clinician-graded adverse events, and was filling a need to ensure that we were capturing symptomatic toxicity directly from patients along domains that were most relevant to their own personal experience. Our goal has been to support the collection and use of PROs to inform interpretation of traditional clinical outcomes and research and to inform care. And real-world evidence generation really begins with the evidence that's obtained in controlled settings. But importantly, while we have participated in the specific tool development that I just mentioned, as in addition to other academically sponsored tool development, our focus has always been to have flexibility in the tools used to fit the research questions, to fit the populations and the designs and the needs of stakeholders. And I really want to comment on the comments that have made throughout the day around the importance of stakeholders and the importance of ensuring that what we are collecting is really fit for purpose. So we work collaboratively across the National Cancer Institute, the National Institutes of Health, with our colleagues at the FDA, at EORTC, with trialists, with other investigators, statisticians, therapy developers. And importantly, we're committed most centrally to understanding and addressing the needs of our most central stakeholders, who are patients, their family and other caregivers, as well as care providers and clinicians. So I want to just close really quickly by saying tremendous appreciation for the presentations today. You started by mentioning, Madeline, that you would have had a different set of presentations perhaps 4 years ago. And it's incredible how much has occurred in the last 4 years. The emphasis on flexibility is not entirely new, but what I'm seeing is a real cultural readiness across different stakeholder groups related to the administration and interpretation of flexible measures, in addition to core and static assessments. Madeline and I had a conversation recently where we talked about the readiness for cultural adaptation and adoption. And the analogy that I love to provide is that of a self-driving car. We had that technology years and years ago, but there was no infrastructure readiness to actually adopt them in our highways and along our roads. But that has changed as the infrastructure and readiness of the community, the population has shifted. And I see the same cultural readiness around flexible assessment and use of PRO information. And you heard a lot of that today. So thank you for including me.
Thank you, Ashley, for your kind and wise words and your support to the workshop today. Jill Bell from AstraZeneca, maybe you would like to share your thank you.
Yes, thank you. So I lead up our COA measurement science team. We're really responsible for developing our strategies across both the early and late oncology portfolio. And, you know, just in reflecting on today, and especially, you know, from what Ashley just said, it's really nice to have listened to the discussion today and the presentations and to really be in a position where what we can do today is different than what we've done historically. Really thinking about, you know, a lot of what Bettina had said earlier today, and really what is relevant to patients? What should we be measuring in our clinical trials? And it really starts with what's appropriate and what's relevant within the specific context of use, because it was mentioned a number of times today already. We can't measure everything in a clinical trial. So really focusing on what are the research questions? What's the opportunity here? And how do we ensure that what we're doing is relevant from a patient perspective? And the opportunity with item lists is to really be able to tailor something for patients, to really understand the patient experience with treatment. I think in addition to that, one of the things we need to consider is to maintain that scientific rigor, which we heard a lot about today. So I think as we work more and more with item lists, one of the things I think we have to think about as we move forward is transparency and how we're selecting those items. And I think there's a lot that goes into that, right? And so we want to make sure that, you know, we're not only asking the relevant questions, but we're documenting the way in which those questions are being considered and included in a clinical setting. I think, you know, a lot of points were touched on today, but the other one I do want to touch on was something that Jim mentioned. I think it's important to think about the end solution, right? And what we're delivering at the end of the day to patients. Because as we develop a measurement strategy on the front end, we also need to think about the end solution and what's in the patient's hands at the end of the day. And this is really critical. So it's not only about asking patients the right questions and also at the right time, which was touched on by a number of individuals today, but also does that solution meet patient needs, right? What are they completing their PROs on? Is it a device that they're able to use, that they're able to navigate? Are we giving them a handheld when it might be an older population or a population with dexterity or vision related issues? Alex, I saw in your presentation you had tablets, which is a really great option, right? Because there's a bigger screen. And so I think we also really need to think about that end solution and what ends up in the patient's hands so that we can ensure we have the quality data at the end of the day.
Thank you. I could not agree more then. My name is Michael Schlichting. I'm the biostatistical HTA and PRO lead at Merck Healthcare. So I say Merck, not Merck. It's a German company, just to keep you aware. And I really love all this presentation. It gives a broad overview of each stakeholder and everything. But on the other hand, so we should think about also, do we over-engineer something here? Yeah. So we address different aspects and have flexible tools. We are on the right way of standardization, even the statistical analysis at CSAQoL IMI. We have standards for protocol, standards for reporting. We are developing standards for these different novel approaches, whatever. But bridging to what we net in the afternoon says, well, we don't have these things for the tumor measurement. So we don't care if the tumor is like a ball or is like a potato or like a ellipse. It doesn't matter. Yeah. But here I think we sometimes may over-engineer something, and this makes it then difficult to assess the robustness. And the robustness, I think, was a critical term that has been used for many people here. And from an industry perspective, we do not know what you mean with robustness. Yeah. So, and I appreciate what Jim mentioned. Well, if we do, let's say, this pro-CTCAE thing in phase 3 trials, we do not know if this is accepted by HTA bodies because it's a limited time of observations, like until end of treatment, but we don't have data thereafter. And I think at the end, we need to map out the data strategy and the research strategy. Maybe we use the item list or the cut system for those optimization questions or those finding questions. Maybe we go to a static questionnaire for phase 3 trials with only limited or selected items that we agree upon, all of these things. I think all things are on the table now. We need to move ahead. We need to leave our comfort zone, because I think otherwise we cannot come to a reasonable decision making for regulatory and HTA decision making at all. So, and I'm concerned that we are too focused somehow to details, and that prevents us to move forward. So we are ready to invest as an industry, but we are uncertain what the incentive is at the end of the day.
Hi, my name is Corneel Koens, and I'm a statistician at the EORTC. I'm also a member of the CISACOL consortium, and I would say I would be pretty happy if I never hear about PRO data ever again. Yeah, I don't want to have this discussion anymore. It's not that I don't value PRO data, but PRO means patient-reported outcome. It simply means it's data that comes directly from a patient. I may be a bit naive. I'm a statistician. I look at numbers on my computer screen. There's no etiquette dangling from the number saying, be careful, this comes from a patient. And I don't understand why we have this workshop about PRO data, like let's put a PRO kit in the corner because he's weird somehow. When we do a clinical trial, we have laboratory data, we have data coming from clinicians, data from performance tests, and you know, in the future, maybe even data from AI algorithms. And we don't have a workshop on, you know, how do we use lab values in a clinical trial? And I just find it a lot of the issues that are addressed can be simply solved by what do we do for the other? What do we do for our other data? You know, why should it somehow come with an asterisk that because it's PRO data? So, for instance, the issue with the flexible and static questionnaires, if we think about imaging data, the way we did imaging in the past from X-rays, MRI, now we have functional MRIs, and who knows what we have in the future. Why can the radiologists, why can they change their tools, but we should not be able to change our tools? I think that doesn't make sense. Why is there a concern that, oh, this patient answered 5 questions and has an outcome, that patient only answered 1 question and has an outcome, and it's the same? That doesn't make sense. Well, you measured 3 target lesions, 4 non-target lesions, and a lot of others, and you said the patient is now progressive after summing up all the diameters and volumes and whatever. That patient had 1 new lesion, is also progressive. It's the same concept. I really don't think, as Michael also said, we may be seriously at danger of overthinking what we do. I think it's time that we say, look, what do we want from a clinical trial? We want to assess the benefit of a treatment. So efficacy endpoints, survival, tumor response, some biomarker, self-reported physical functioning. Let's put PRO data in that column, not with an asterisk, not in a separate chapter. Safety data, toxicity ratings, dose modifications due to adverse events, self-reported treatment burden, goes into that column. Too much we see protocols where PRO is in an appendix or a separate chapter or has a separate section. The SAP is developed with PRO as separate. Let's put it all together. Let's just consider PRO data as data, like laboratory data is data, clinician data is data. And a lot of the issues, it's subjective. As Paul Kloot said, are we kidding ourselves if we say clinicians are not subjective? But we know what clinical data means. Body weight as an objective outcome. Okay, 65 kilo, what does it mean? High, low? It's the same thing. You need context, you need to know what it is, and we can learn that. So I think, with these new instruments, etc., let's just see them for what they are, tools that we can use, and let's just talk about PRO data for how we want to use them and not where they come from. And so, yeah, I would be very happy if we can stop talking about PRO data.
So it's very hard to ask questions after a very interesting, after everything that our panelists have said. So, I mean, Chantal and I have planned questions, but then after everything that Ashley has said online and about the issues that we all have heard, and after going after what Cornel's rant is about why we have different standards for PROs compared to clinical endpoints. So anyway, but we will ask them anyway, because this is about novel measurements. And I think, we take for granted that here, at least in this room, a lot of us are very familiar with PROs, right? But there are a lot of people out there who are not familiar with PROs, or smaller industries, SMEs, or beginning groups or clinicians. So my question to Hans, to Mons, to Ashley, or to Jill for my measurement strategy people, when earlier, like Paul Kloot said in his statement, thoughtful consideration of the use of static and item libraries, what would your advice be when people start off in developing their trials with a PRO endpoint? What does that mean when you say thoughtful use of static questionnaires and item libraries? How should they do that? Where should they start? Does anyone of you want to comment on that? Yes, Mons?
I think when people come to me to ask which questionnaires they should use, I always send them back and say, please explain why you are doing this study. And I think that's where we are spending too little time, and where we are not having very clear. The only instruction is to think well, pose your research questions, and review the literature and talk to the stakeholders. And I think that's an area where we could do better. And this lack of transparency about which outcomes are used in similar studies of similar interventions, for example, it's very difficult to get an overview of what did all the other people doing the same kind of studies, and which are the top 10 side effects measured for different treatments. Everybody is expected to review PubMed and then come out with a response. But I think, and we've been discussing that when we started the new strategy, that having some kind of repository of either of reviews of adverse events of treatments, or experience from previous trials, or some filters you could put into clinicaltrials.gov, where you could actually say that all people who would study a particular drug should relate to what did the others do. And then when they present this overview of what did the previous 20 trials do, then you have a very good basis for saying we do the same, or we exclude something. But then you have a transparency, and you might even add, based on your curious work, saying that we need to include these 3 others, and then the next one could consider this. It's just maybe it's not possible, but I think that's an area that has not really got any framework that is generally accepted.
Thank you so much, Mons. Ashley also has her hand raised. Ashley?
Yes, hi, thank you. I think I want to augment a little bit of what Mogen's just indicated and extend what we've heard previously. The decisions need to be a balance of precision, breadth, feasibility, and value. And that's not going to be seen the same way by all stakeholders, particularly the value piece and the feasibility piece. So of course, we have to start, as Mogen was just commenting, with the question. What's the research question and what is the intention for the information that's collected? And then when thinking about a specific tool, there are many investigators who've become very comfortable with specific tools, either due to their long-term use or availability via their institution or in the community that they're comparing to, or availability based on other platforms. So what's critical is for investigators to understand the benefits and limitations of any instrument in terms of the information that's provided. So the content and again, specificity and how well it answers the research question, but also how easy it is to complete, whether the patient and provider see value. Because if they don't, and this was mentioned earlier, and thank you so much for our patient stakeholders, is, completion rates are going to be low if people don't see a value, and not just for the common good. So there's also need to think about complementing other data that is collected. Maybe it's other clinical data, and to parsimoniously provide the information that's of relevance, particularly if you're thinking about it in a regulatory context, to decision makers and who's evaluating that information. So those are some of the considerations that I think need to be put on the table early and thought about throughout the process when determining which tools to use. So I'll stop there.
Hans, and then Jill.
I would like to add choosing the right item for from a design perspective of a questionnaire. I think you first need to look at the situation, what the impact is for the patients. I think we've had once been involved in a clinical trial from the sideline as reviewers, but also in the EMA process later on, where we had a drug where there were some issues with the eyes, the ocular situation, and the report said, well, we have grade 1, 2, 3, etc. You can quantify that, and the in the report was asked to the clinicians what the impact was, and they said it's manageable. So that's not that long ago. We are so long busy with PRO data. So I think when you really want to know what the context is, and that's what we gave back also to the company, also to the EMA at the time, is the context is that when your eyes are blurry, you cannot drive your car, or is it dangerous for walking the stairs, for example? That is the context you need to have as a patient to make a decision in the end to decide in a shared decision environment for this treatment or not. So manageable is thinking from the clinician or the hospital perspective, but you need to have those items, those extra items, those PRO items in the questionnaire where you really understand the context of this kind of reporting.
So I'll be brief because I think a lot's been said, but one of the things I think that's really important to consider, in addition to the question and the tools and what we're measuring, is when we're asking the question. So it's not just about making sure we're asking the relevant questions, but it's also asking them at the right time. And that's so important and so critical because it has to be tied back to your research question and obviously the endpoint. So we might be asking some questions daily. Other questions might be weekly, or some might even be cycle-based. So you really want to think about that timing of your PRO assessment because it actually does matter. And so that's just the one piece I'll add that I think is really important upfront when you're thinking about your measurement strategy.
All right, thank you so much. We'll probably create a checklist after this so that we can put it in the report based on all of your comments. All right, Chantal.
Yes, I think it was also important to kind of address a methodological question, and the reason is actually this, that we here at EMA, we do acknowledge that the PRO world is moving forward, and I think it was already addressed by my EMA colleagues like Carla Torre, who reflected on the challenges and limitations and potentially improvements or action points that could come from that. Also, my colleague Juan Garcia about the PhD reflection paper on patient experience data, which is something, a signal that the EMA is kind of moving forward, also accepting or including PROs. But we also have not only like high-level principles and reflections that we should and could, but we also have made initiatives already like hardcore in our guidance document where we do embrace patient-reported outcomes. And I would like to highlight actually a public consultation that is currently on our EMA website. So it's there for everybody to have a look at. And it's a guide for assessors when they assess marketing applications. And we have included EMA a chapter, and I'm sorry, Cornel, it is a chapter on patient-reported outcomes. But at least it's there. And I think it's very nice that we, by including these sections, we kind of allow more patient-reported outcomes to be reported, and that they don't find or lose their sense in clinical or lab data. And one of the sentences that is mentioned there as a guidance to the clinical assessors is that PROs needs to be clinically relevant, statistically robust, and informative so that they can be included in the summary, right? And no other sections, if not, they will not be reported. So I think it's really an action point for the PRO community to kind of look at this, at that section, and to kind of provide content and guidance to this, to these elements of what is statistically robust or what is informative. And I do understand that CISCOL IMI is contributing extensively and hugely to addressing these questions and to provide content and standards to these elements. But I wanted to come back a little bit on the term clinically relevant, which is measured here. And I come back to the presentation of Johannes earlier, where you say it's misused or what is a clinical difference, etc. And I wanted to kind of understand a little bit from the methodologist, because it's also part of CISCOL IMI, how they think the term clinical relevance should be further elaborated, so it could be easily interpreted and not misused. If somebody wants to answer it. Yes, thanks.
Okay, so I think there are many problems with the term itself and with the methodology underlying this. Just to highlight a few of the challenges we're facing. So as said earlier, like a big challenge is that there's like currently little distinction, I would say, in literature when it comes to differentiating patient level thresholds and group level thresholds. But it's like very commonly, I feel like confused. It becomes way more clear for other clinical outcomes, and another reason why we should maybe not separate so much PROs from other outcomes. And the usual example I use is like, let's say, blood pressure, where it's like, again, a very abstract metric, like a millimeter mercury, very abstract to me. And we have like kind of established thresholds when we evaluate therapies for hypertension. And if you look into the literature, it's a common definition for responders in these trials that like 20 millimeter change would be considered clinically important for a patient. But at the same time, it's very clear to us that we cannot expect a difference between trial arms to be of that size. Usually, like they will conclude like clinically relevant improvements if the difference between trial arms is something like 4 to 5 millimeters. So it's very obvious in that regard. But the same thing applies to PROs, where it gets completely blurry, I would say, and people very often use like this, I would say, this 20 threshold to interpret treatment with difference between trial arms, resulting in the very common conclusion there's no difference clinically between the treatments. So like that's one major aspect I feel. Then when it comes to terminology, maybe to highlight that as well, and like also like kind of promote the work of CISCOL, there's lots of confusion when it comes to the various different terms being used: minimal important difference, clinically meaningful difference, clinically important change, it goes on for quite a while, I guess. And very often these terms mean the same concept. Sometimes they don't. Very often they do mean the same concept. Then there's quite a variety of definitions for these terms that are often not fitting the context in which such thresholds are used. And on top of that, we have like quite different methodologies on how we establish these thresholds. And I would like strongly advocate not to look so much at the terms that are being used to label this, but really have a close look at how these thresholds have been established, because the methodology, how you establish this, will very strongly determine what the threshold actually means. I'll give a quick example, then I stop. Is, for example, it might differ if your threshold reflects a change in a symptom that a patient is just able to notice, which is a common approach, compared to a change in a symptom that equals like a major improvement in performance status, equaling one category change on the ECOG status. Just like as a quick thought.
Maybe we might also get some feedback from statisticians who really incorporate quality of life as endpoints. And I'm looking at potentially studies that are done at the EORTC or not, like, because as far as I understand, I only know about the 10-point, and I just wonder how, is it the 10-point in every case, everywhere, or, and then there must also be a difference between improvement and deterioration. So there's a lot of, or are EORTC statisticians just using 10 points? That's my question. Thank you.
Oh my God.
Well, I had just a quick thought, because when we speak about clinically relevance, we really think of from a physician perspective, right? Making a different treatment decision or a different concern. When you think about PRO, it's about what is a clinically, or what is a meaningful difference to patients. We have quite a methodology to explore that. That's called patient preference studies. So I'm just thinking you could use patient preference studies to really determine what matters to patients as a threshold. Just a thought.
So, okay, can I go further? So just taking this MID topic as a perfect example of what should we do from today, that is reaching some kind of agreement. Because, for example, MIDs is really an issue when you want to design a study. If you're going to consider the PRO as a key secondary endpoint, considering 10 points or 15 points or whatever points has an impact on sample size. So from an industry perspective, so I work in industry, but I have a lot of experience before in academia. So I know both sides. What we will need is some kind of agreement on a core set of minimum requirements between not only, between different stakeholders, that I would say FDA, EMA, HTAs, because otherwise I fear that, when we have to design a study with PROs, we have to take into account so many different perspectives. And I agree there could not be a standard for everything, but at least a core set of standards. Or also about terminology, as Johannes was underlying before. That would be really, really helpful. Also in the interest of patients, because otherwise a lot of PRO data would be thrown away and there would be no advantage for anyone, and at least for patients, I would say. Thank you.
Maybe one final comment from Cornel, and then we would like to close this session. Thank you.
Okay, so very, very quickly, what do we do for the other endpoints? So when is a survival difference a relevant survival difference? Is it 3 months, 3 weeks, 3 days, 3 years, 3 decades? Honestly, it's the same question. It's relevant when it's relevant, and it may differ from situation to situation. You will have, I think, the application, like Johannes said, with the individual thresholds is very helpful for a sort of safety toxicity reporting. But let's say we want to design a trial where we expect an improvement in survival, but we don't want to reduce patients' quality of life. So it's non-inferiority for quality of life. I would say that the margin, the price you're willing to pay in quality of life will very much depend on what you stand to gain in your survival. So I don't think you have a fixed number there. And that's when you make the decision on a population level. Per patient, a patient, when you have the consultation and you have to make the decision for that patient, it may still be quite different. So while you may have a general treatment burden, general score that works for, regulatory HTA bodies because it works on a population level, for a clinician and an individual patient, you will want to see, but what are the actual symptoms here that matter, and how do patients who have lived through this, how do they rate them? So it's basically the same as we use the other endpoints. So in the SMPC assessment guidance, when you say PRO endpoints need to be statistically robust and clinically relevant, that's only for PRO endpoints. The clinicals don't have to be statistically robust. Why is that sentence there? Take it out.
They do, but this was related to
have to fulfill the criteria or none of them. Don't patronize PRO data.
Actually, this is public consultation, so you're welcome to comment on the text.
I'm going to go online and make the comment right now. Thank you.
But we will close the session, and I thank you.
It would help Cornel if there's a chapter on laboratory measures as well. Is there or not?
Yeah. Well, the laboratory measures do not easily also get there. So for me, the task to wrap this up. I have 15 minutes for that, they said, and I think most of you are very tired. I think, first of all, a big thanks to Caroline and Madeline for really pushing on. And Cornel, you sort of hijacked the meeting in a way where we need to respond to an individual here. But I believe, and I'm not sure how many people are out there, but there's a lot of people also online, and there's a big interest. Obviously, PROs, and when I talk about it with some of my colleagues, they say pain is a PRO. So we have it for a very long time. But I believe we are in a different setting. We're looking at more the holistic also concept of capturing what the patient feels and functions. And I think dealing with that is something that we try to do. As regulators, we are by nature a bit conservative. We need to see what the plans are. And I think, gosh, now I forget your name, Alexandra, you fantastically presented how you can, in the best way, have a fixed data set and add to that something that is flexible to the needs. And as regulators, we are experiencing so many different diseases, also outside of oncology, where it is difficult to deal. And the same applies obviously to HTA, where you have to deal with this emerging set of different diseases and drugs that we are treating patients with, and also becoming increasingly complex. So indeed, there's a lot to say that there is an absolute interest in having these kind of tools. Then, though, is the issue, and there is the conservative part, is that somebody needs to demonstrate, and that's usually the industry to us, that their drug does something on this type of outcome. And that and there I liked the start, and now I'm going to go back to the start of the meeting, where both Pierre and Jaap, sitting next to me, really illustrated the different kind of trajectories patients go through, and what can happen if you look at similar kind of populations in a trial setting, but have complete different outcomes because you measure at different time points and you haven't made up what the estimate exactly is. And here's that horrible word again, which we invented a number of years ago because we as regulators have to rubber stamp what somebody says you have shown to be doing with your drug. And it's very dependent on how you fix that. That estimate has to take into consideration what the patients really, or actually the users of the drug really can understand that the drug does for them. And I think that is where very clear also today again was talked about the different perspectives where you can look at. And that is the whole patient journey. And then I think that is something that is also clearly important. Question is how to go back and translate that to a trial setting and to something that can be put forward to a regulator or an HTA to make a basic decision on, or other stakeholders more downstream. So then I think I come to the middle part of the meeting, where we had a lot of discussion on how to define, let's say, what we want to do and how can we make more harmonized approaches to understand these data, and also how to deal with that. What are the expectations on the side of regulators and HTA? What are the expectations on the side, or what can be done in that trial to improve that? And I think there is clearly an important message. And I think the statisticians are very clear about make sure that you define beforehand what you want to do. Harmonize the development of the PRO tools. And I believe that the EORTC people have done a very good job in setting that up, wanting how to do that, but also indeed it's context dependent. Think about language and the cultural setting where you're doing it, but also plan for those different users that are there. And so indeed, then think of, okay, what is exactly what we want to show with the trial and with the study. And there I think it was an interesting discussion that we had around precision. I think Ashley made that comment, precision, breadth and feasibility of the data, where it is important that we can try and capture everything, but maybe as this is still somewhat newish, we should really be careful in how to define what we can really show realistically in the trial. What can we also collect in the trial? And then there are interesting discussions that Beate Wiesler said, well, we need to have that clear precision, where the neighbor who left from the other HTA said, well, but we have a different approach and we can deal with the uncertainty, but at least the planning and the thinking through what are the kind of data that you want to demonstrate and how do you do it? So it is fantastic if we can have these whole patient journeys. And I think Douwe Postma showed this very beautiful picture where you saw what happens if you take this drug. You have this progression-free survival with or without toxicity. What is happening in that next interim? But I believe you can also interpret these pictures already quite well if you have the uncertainty that is still left there. So I think we should not throw away the baby with the bathwater and trying to solve all issues at one time. So I think that's an important aspect. I think also two other things that were mentioned is we may want to collect more information than you can feasibly do in a trial. And I think another aspect is that we obviously talk about what can we gain from real world data. And I think that's one of my also other interests. So I think it's good to mention at least that might be important, what is done there and what is feasible in that setting. And I think finally there was a comment made around, okay, so what, and then the room exploded, I think, when we talked about the minimal clinical important difference. And yes, we should have had another day of meeting and tonight lots of drinks and go on tomorrow. But I think one of the interesting points is there that it on one hand looks very much like blood pressure and you can measure that. Also, I can tell you in blood pressure, I come from the cardiometabolic background, there has been a lot of discussion around responders and how many responders there are in the trial to also really contextualize what you actually see in the average effect in the trial. But clearly that is there. And I think the other aspect that somebody there on that side mentioned is that you can obviously ask the patient and the patient preference studies that can now also be used to really understand what the perspective of the patient is on what they want to see. And finally, I think, and that is Francisco's doing also, he said to me, Peter, make sure that at the end of the meeting, we really conclude that we need to go forward. We need to build a smaller group. I think that continues the discussion that we had today on, well, developing PRO into sort of normal numbers, into normal data. But at this stage, to really mature what we have addressed, let's say, or identified as problems with some tangible solutions, but also really to move on. And I think that's in good hands with the colleagues here from EORTC and the EMA Secretariat, but I believe that is really important. And it looks very much like what we did in the Patient Registry Initiative. We talk always to industry as regulators. Industry talks to in this case, the academic PRO developers, but there needs also to be that crosstalk between the regulator and the academics. And I think that's a fantastic start today. Hope to see that continued. And with that, I thank you all for your endurance and stamina to stay here. I'm pretty sure some of you will have very good after discussions, and I'm pretty sure we will, in different groups, meet again in the future. Thank you for your attention.